live? Hi kids. Hello. Hi everyone. Are we live? Hi kids, am I audible? Yes. Am I audible? Hi everyone. Kindly let me know, am I audible? Good evening. Welcome to Unacademy NEET English. I am your biology educator Ambika Sharma. I hope I am clearly audible and visible to you all. So, welcome to this amazing session. Today we are going to target the biological classification. It will be a one shot video, but here in this video we will move from basic to the advanced level. So, it's okay, it's completely fine. Even if you are doing this chapter for the first time, you will be able to understand each and everything in a very smooth flow. And content, obviously there is no doubt in it, right bache? You will get the best content, isn't it? And I hope you guys have also attended my last session, that is on the living word. So in a sequence we are going to complete the chapters. So after biological classification we will go for the plant kingdom and the animal kingdom, and there will be the regular practice of the questions as well. Okay. Chapter will be, we are going to do it in detail guys. So here you people can see, yes, here you all can see, this is the description box, of course here I am teaching. So you guys can see this button here. So quickly everyone just hit the like button here, subscribe the channel here, and here in the description box you guys can see, right, if you want to be the part of our batch, we are teaching on Unacademy platform, and the name of the batch is the NEET Energized batch, and it is a re-entry point for the NEET Avengers batch. And trust me, we are getting amazing feedback from the students of NEET Avengers batch. They are really loving it, they are enjoying it. Okay, so what you have to do, you just need to click here, which is a link for the NEET Energized batch, and kids, here you need to add your mobile number or the email, whatever you want. So after the coupon code you will get this batch at this price. Okay, so it's a very reasonable amount, and 11th and 12th syllabus will be completed. Right now we are doing the 11th syllabus, then we will start the class 12th syllabus, and time to time you used to get the strategy based session, the motivational session, the detailed question practice sessions, and many more things are there in this particular batch. So if you have joined it, very good. If not, be the part of this batch as soon as possible. Now let's come to this particular chapter, that is our biological classification. So kids, what do you know about this particular chapter? Tell me, what do you people know about this particular chapter? Whatever you know about this particular chapter, just let me know in the chat section from your previous knowledge that ma'am, this is the meaning of biological classification and this is what we are going to read in this particular chapter. And first of all, even if you are attending our session for the first time, be comfortable, take it easy. It's a long marathon, right? So I want to take this session for the duration will be, right, the duration will be 3 to 4 hours. Rest, it depends upon your response. Okay, it depends upon your response. And moreover kids, we will go in detail. So even if required, I will take one extra session for it as well. Isn't it? Ultimately our motto is to clear the concept. So now let's start the session, and the chapter name is biological classification. So I am going to relate it with the last chapter that we have already started. So bache, you know the one thing, that is in the living word, right? We have already completed this chapter in NEET Conquer batch, right? So when we talk about the living word, here we talk about the taxonomy, isn't it? Here we talk about the taxonomy. So what exactly is the taxonomy? Can you tell me? Anyone here? Can you tell me? Kaushal. Yes, bache, we will complete the syllabus, but here, right now, please focus here. Right now, please focus here. After some time, I will see your charts, I will revert, but now, just answer my question, that when we talk about the taxonomy, what exactly is the meaning of taxonomy? What do you know about the word taxonomy? Classification. What else? Basically, taxonomy is also the branch of biology. This is what we know. right? It is also the branch of biology. It is the branch of biology in which we will, right, study the principles, the procedures which are used for the identification, for the classification, for the nomenclature, right, or basically identification, nomenclature, and for the classification. What should be the sequence? What should be the sequence? Can you tell me? What should be the sequence? So basically, you know that we have to identify something, we will classify it, we will give it a name. So this is what we study in the taxonomy, bache. And in the taxonomy, the basic to taxonomy is, right, the basic to taxonomy is what? It is chicken. This is the trick that I have given you in the last class. What was the trick that I have given you? It is the chicken. So what is the meaning of chicken, bacha log? What is the meaning of chicken? CH stands for characterization, I stands for identification, C stands for classification, N stands for nomenclature. This is what we have covered in the last class. Now you must be thinking that, ma'am, why are you teaching the topic of last session? Because we are going to, right, connect two things together. Right, bache, and this is how we are going to learn in these particular batches. It's not like that biological classification finish, now we will revise it later. No, we will relate everything. Right, this entire unit we are going to study it in that way. So what I just said, for a quick revision, everyone just note it down, take a note of it, that when it is the taxonomy, so what are the basic to taxonomy? Guys, I want to see that in the chat section. What are the basic to taxonomy? That is CHICN. CHICN. Right, CH stands for characterization, I stands for identification, I stands for identification, C stands for classification, N stands for nomenclature, and here we will talk about certain set of rules which we use to classify the organism. So now the word here is classification. Why? Why this classification is important? What is the meaning of classification? I'll give you a very simple example, right? A very simple example from your day-to-day life. Even if you, whenever you look around, especially in your home, right? If you are living in hostel, then I don't know whether it is applicable to you or not. But yes, if you are living in your home, then definitely it is applicable, right? You go to your kitchen. You know that your mom has arranged everything properly, isn't it? Your mom has arranged everything properly. Right, your mom, there will be a box where the sugar will be placed, right, where the tea will be placed, right, like this. There will be one almirah for the pulses, for the spices, like this, right. Let's take the example of your almirah. How do you arrange the clothes? In your wardrobe, in your almirah, in your cupboard, how do you arrange your clothes? How? Right, like there are two ways. You can put all the formals together, you can put all the casuals together, or you can keep your t-shirts on one side, you guys can put your jeans, your bottom wear to another side. This is how we arrange the things. Why? Because it is going to make the things easy, right? We can use them easily otherwise. Now just imagine, you are going to a library. My best example is library. You are going to a library and you have decided today you are going to read the entire physics. Right, just imagine it in this way. You people are going to the library and you have decided today I am going to finish the entire class 11th physics. Right, no matter what. Right, next day I am going to ask question from the Shreyas sir. Arey, Shreyas sir, you are teaching us this. I have already completed it. Now I will ask you the question. Just imagine, you are going to library and you are like, so many books are there. You don't know which book is for the physics, which is for the chemistry, which is for the biology. So firstly, you are going to library, you are searching for the book and then you will study. Right, obviously, in that time period, you will be like, no, I don't want to study now. I'll go home, I'll take the rest. Right, I'll go home, I'll take the rest. I'm not interested in studying. It's not a good library. Firstly, I have to go there, I have to find out the book, then I have to study. Oh, no, I'm not going to do that. This is a usual thing, okay. Now in the library, when we know there is a proper section of the physics, there is a proper section for modern physics, for this physics, for that physics, okay, you will go, you will take out your book and you will start studying because the systematic arrangement is there. Right, the systematic arrangement is there. That is why. Now, when you talk about the living organism, guys, so you know that, right, when you talk about the living organisms, you know that more than 1.5 million, more than 1.5 million, or you can say that in the range of 1.7 to 1.8 million different species are there on planet Earth. You know about it, isn't it, guys? Isn't it, guys? So more than 1.5 million species are there on this planet Earth. And when we talk about these species, we are just talking about the eukaryotes. We are just talking about the cells which are having the nucleus. We are not even talking about the prokaryotes. Are you getting my point? We are not even talking about the prokaryotes. So just imagine on this planet Earth, if we have so many species, so it is not possible for us to go and to check one species and to study about it, to another species and to study about it, to another species and to study about it like this. We cannot do that, right? So now if all the things will be in a systematic manner, right, so what will we do? We will study about the one thing, we will get to know about the so many things. So when it comes to the classification, actually it's a systematic arrangement. Okay, in simple words, if I have to explain you this part, so what is it? It is a kind of systematic arrangement. Systematic arrangement where on the basis of certain similarities, right, where on the basis of certain similarities and dissimilarities, on the basis of certain similarities and dissimilarities, we are going to classify the organisms. We are going to put them in a different groups, isn't it? We are going to put them in a different groups, right? Is that clear? Is that clear? This is what you can understand now, right? It is very easy. Now what is the next point here, kids? What is the next point here? This is the meaning of classification. Now when we classify the organism, when it comes to the classification of the organisms, you even know one more thing, bache, right? We have the certain categories. Can you name the 7 broad categories? Yes, bacha. Can you name the 7 broad categories? What am I saying? We are talking about the classification. We are talking about the classification here. Yes or no? Tell me quickly, everyone. Yes or no? Just allow me a minute. Okay? Okay, now it's fine. Okay? So, tell me about that 7 broad categories. I want to see the answer in the chat section, guys. I really want to see the answers in the chat section. Yes, all of you. So, when you talk about the classification, you know that there is an hierarchy. The taxonomical hierarchy is there and 7 broad categories are present. Right? 7 broad categories are present. And that 7 broad categories are, right, so keep pot clean or family gets sick, right? It will be keep pot clean or family gets sick. Are you getting my point? Yes, bache. Keep pot clean or family gets sick means kingdom, right? Kingdom, then comes the phylum, then comes the class, then comes the order, family, genus and species. These are the 7 broad categories that we have, bache. And out of it, right, when you talk about the kingdom, it is the highest category, right? It is the highest category. And kids, it is also known as taxon. Taxon is singular, taxa is for plural. Right, this is basically the rank, this is basically the hierarchy, this is basically the rank, this is basically the category. You can even use the word taxon for it. Taxon is for the singular, taxa is for the plural. This is what we know, guys. This is what we know, yes or no? So, this is about the kingdom. So, highest taxon, highest category is your kingdom, and when it comes to the lowest one, that is will be the species. So, right. So, these are the 7 broad categories that we have. These are the 7 broad categories that we have, right? And we have studied that in the last chapter. If you want to revise it in detail, you guys can check the living board. Now, in these categories, as per the similarities, as per the dissimilarities, we are going to arrange the organisms. But now, here in this chapter of biological classification, what are we going to start, bache? Firstly, now we understood the meaning of classification. We know that when it is the biological classification, we are talking about the living organisms. We have to arrange the living organisms because their number is too high. So now we have to arrange it. There is the hierarchy there, and now we have to talk about these kingdoms. So up to this part all clear, guys? Up to this part all clear? Now what is the another part? Now first of all, right, for the very first time, you know who tried to put things properly, who tried to arrange it properly? Do you know the name? Do you know the name? Yes, bacha log? Do you know the name? Bache, I will provide you the notes in the Telegram group. Last time also I forgot na. Don't worry, this time I'll provide you. Don't worry about that. Okay, don't worry about that. There is nothing to worry about. Okay, there is nothing to worry about. Okay, I'm just... Tell me. Who started it for the first time? And guys, I really need to fix it. Just now I hope it's fixed. For the first time, yes? Of course, for the first time, Aristotle tried it. Who tried it? Aristotle. Who is Aristotle? He is the father of biology and also he is the father of zoology, isn't it? Do you know about Aristotle? He is the father of biology, he is the father of zoology. So bache, for the very first time, the scientific attempt to arrange the organisms, right? He made that scientific attempt, right? Basically the first scientific attempt, he made it. So he is the father of biology as well, he is the father of zoology. What is the name? The name is Aristotle. So Aristotle tried to arrange the organisms, right? Who tried to arrange the organisms? It's the Aristotle who tried to arrange the organisms for the first time. Now what he did, he was like, his way was very simple. Okay, his way was very simple. Even I have added some slides, some notes there, right? For your quick revision, you will get to know about it. But as of now, just focus here. So Aristotle was like, he checked the morphological character. So how he tried to arrange the organisms? On the basis of morphological characters, right? Morphological characters. Kids, can you tell me about the morphological characters? What do you understand by the word morphological characters? Anyone in the class? Morphological. So when the word is morph means form, right? Logical, morphology, logy means study. So basically the outer form will be studied here, right? The outer characters which we can see very easily. You can just look at a plant and you can say this plant is not so tall, this plant is short, this plant is like this plant is like that. So when it comes to the morphology, yes, the characters that you people can see, the external characters, of course, right? Here what are we discussing? We are talking about the external characters here, right? Outer appearance, you are very right. So on that basis, what he did, he tried to, so when it comes to the plants, right? When it comes to the plants, what he did on the basis of their size, like some are very small, herbaceous, herbs, shrubs, right? And then trees, isn't it? Herb, shrubs, trees or herb, shrubs, plant. How did he arrange? Can you tell me? Herb, shrub, trees or herb, shrub, plants. Like this, he has arranged the very small herbaceous type of plants, right? Some are bushy, shrubs, then some are taller, the trees are there. In the case of plants, that's how he arranged. And when it comes to the animals, it was very simple. He used two words. One is anima, A. And another is enema, right? A and E. What is the key word here, bache? One is A, another is E. So A means without. What is the meaning of A? A means without. Without what? Without RBCs, or you can say that without red blood. Without red blood. So the organism, in the case of organisms, in the case of animals, what he did, right, on the basis of presence of red blood group, red blood and without red blood, that is how he divided. So anima means without red blood, without RBCs, and enema means with red blood. So that is how he categorized the, he categorized the living organism. But do you think it's a proper way? Do you think it is the proper way, guys? And we have so many students and number of likes are quite less. So guys, quickly hit the like button, and if you are new to this channel, subscribe this channel, because here we are going to provide you the quality content with all the updates that are required for your NEET examination. Even we used to guide you time to time to tell you that this particular course is good, this particular course is better for you other than NEET also, right? So for such guidance, please support this channel. Do hit the subscribe button as well. And as I said, you guys can join us on Unacademy platform. You have to join the NEET Energize batch for that. Done, bache? So this is what you need to keep in your head, right? So obviously Aristotle was the first one who tried it. It was a scientific attempt, but it is not the appropriate way. That by just looking at plant, you can say okay, on the basis of its size, divide it. Or in the case of animals, we cannot. And we have variety of animals, we have variety of plants. So this is not a good, this is not an appropriate way, right? So this was the first attempt, right? So in this continuation, we will learn everything. So here you guys can see a complete, right, a complete flow chart that we this is what we need to do in this particular chapter, that is biological classification. Guys, chapter is lengthy, right? So this is what we need to discuss. Like, we will start with the classification system. Now I have just given you the introduction. Now we are going to start this particular part, the classification system, 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, 6 kingdom. This is what we need to discuss. Then individually we will be talking about the Monera, Protista and fungi in this chapter. And finally, the exception of this kingdom, that is viruses, viroids, prions and lichens. Okay, so this is what we need to discuss in this particular chapter. The chapter is important. It is important to understand the diversity. It is important to understand the basics of the diversity. Clear, bache? Clear? So here you guys can see, so the first attempt was made by Mr. Aristotle. Who made the first attempt? Aristotle, father of biology, father of zoology. Now we will talk about the 2 kingdom classification, the 3, the 4, the 5 and the 6. Guys, you can take the screenshot of it, and moreover I will share the telegram in my I will share the PDF in my telegram group. Now let's start with the Aristotle. I told you already, plants, herbs, shrubs and trees, animals, anima and anima. So here, if you ever get confused, just focus on this word A. A means without, right? A means without. So A is without RBCs, without red blood cells. Clear, bache? Clear? So now come to the 2 kingdom classification system. So you know the name of the scientist. You know the name of the scientist who gave the 2 kingdom classification system. Bache, you all know about him. 2 kingdom classification, right? That's 2 kingdom classification. You know about it, yes? So obviously it is given by Carolus Linnaeus, right? Carolus Linnaeus. He is also known as the father of taxonomy, isn't it, bache? He is also known as the father of taxonomy. Who's he? He is also known as the father of taxonomy. We have studied about him in the chapter Living Word, right? We know that binomial nomenclature was established by him, right? Binomial nomenclature was established by him. We have already studied that. Now, bache, Carl Linnaeus, he gave that kingdom. He used that term kingdom, right? You must be thinking that, ma'am, if Aristotle has started it, then why for the kingdoms we talk about the Carolus Linnaeus first, right? Why do we discuss the Carolus Linnaeus first? The point is, bache, because he, for the first time, he used the word kingdom. So when you talk about the Carolus Linnaeus, he has given the 2 kingdom classification system, and he selected something very basic. Do you know that? For the 2 kingdom classification system, he opted, you know, very simple way. He was like, on the basis of certain character, he was like, 2 kingdoms he has used. He had used, one is plantae, another is animalia, that's all. He divided all the living organisms as plants or the animals, right, bache? As plants or the animals. Now when you talk about that characters, what he selected, what he opted, right? For these are the features. First is cell wall. The very first character is cell wall. He was like that if the organisms, they are having the cell wall, means they will be included in, they are the part of plantae. Okay, okay, simple, no tension at all, right? No tension at all. You must be thinking that, ma'am, we should accept the Carolus Linnaeus kingdom system, isn't it? Isn't it? It's very easy. If you have the cell wall, come in the kingdom plantae. If you don't have the cell wall, go in the kingdom animalia. It's as simple as that, isn't it? It's as simple as that. But now, you know, scientists, they studied a lot, they studied this, they studied that, and now they're like, no, 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 we have 5 kingdom classification, we have 6 kingdom classification. Hai na? Now you have to remember so many names. But initially it was all simple. Plants, animals, isn't it? So see the features here. So if cell wall is there, cell wall is present, welcome in the plant kingdom. Cell wall is absent, welcome in the animal kingdom. Simple. Now come to the locomotion. If you cannot move, if you are immobile, not able to move, come to the plant side. If you can move, go to the animal side. Simple. Yes or no, that's all. Okay, so if you can respond to external stimuli but your response is quite slow, go to the plant kingdom. But if your response is fast, go to the animal kingdom. And when contractile system, right, in the case of contractile system, obviously contractile system that can contract, that is present in the animals, not in the case of plants. So these are the characters that he has selected, he had selected. Are you getting my point? Are you getting my point? So it was very simple when you talk about what? When you talk about the 2 kingdom classification system. Hai na? Hai na? So Carol Linnaeus was like, yeah, I will make it very simple for all of you. I will make it very simple for all of you. So now, what are the contractile, which is able to being contract? You know, na, contract, relax, contract, relax. In the case of actin protein also we discussed the word, it's a contractile protein. It can undergo contraction, it can contract, right? It can decrease its size and then come back to the original size. Clear? So this is it. Now when you talk about this particular system, guys, even we will revise it from NCERT as well. It's not like that we are going to leave NCERT, not at all. We will revise it from NCERT, but right now just listen to me, okay? Then we will come to the NCERT. So now the next point is, okay, fine. Linnaeus was like, see, the things are so simple. I don't know why you people are making it complicated, right? So animal, plant, animal, plant, animal, plant. But it was not like that now, because there are many objectional things also. Let's take the example of euglena. Let's take the example of euglena. When we talk about, right, when we talk about the euglena, you know that it has characters of both, right, animals as well as plants, isn't it? It has the character of both the organisms, animals as well as plants. It's a connecting link. So where to place such organisms which are showing the characters of both? So it was a kind of question mark, yes or no? Guys, the chapter is lengthy, but it is not boring, huh? It is literally not boring. We will interact in between and we are going to make it very easy. Okay, what say everyone? What say everyone? Right, we are going to interact and we are going to make it very easy. What say? So I want to see the energy in the chat section. Quick, quick, quick. I literally want to see the energy in the chat section. That, okay, ma'am, we will make it easy. Right. So now, because of Carl Linnaeus, the Chlamydomonas, right, the Chlamydomonas and Spirogyra, Chlamydomonas and Spirogyra, they were placed together. Now, if I talk about these days, you know that Chlamydomonas is what? Chlamydomonas and Spirogyra, because they both are having cell wall, so he kept it in the kingdom plantae. But it is not like that. You know that when it comes to the Chlamydomonas, it's unicellular, right? It's unicellular. When it comes to the Spirogyra, Spirogyra is multicellular. So he ignored such characters. He ignored such characters. Are you getting my point? He ignored such characters. Right, bache? When it comes to the Carl Linnaeus, are you getting my point? Are you getting my point? Yes or no? Yes or no? Yes? Yes, bache? And now if I talk about the fungus, they didn't, right? If I talk about the fungi, the fungus, so fungi, you know that fungi is heterotrophic. And if I talk about the fungus, fungus is also having the characters just like animals or plants too, right? Right. So what about these type of organisms? So when it comes to the two kingdom classification system, so we can say that Carl Linnaeus, he didn't pay attention on the unicellular and the multicellular organism, right? Guys, do make the notes of it, right? Right. Although we have the NCERT, but still try to make the short notes. Please try to make the short notes, because whatever extra is required, I'm going to tell you in this particular session. So the things are going to be very easy for you. So it's a request. Please focus here. Okay. Please focus here. So what are we saying? We are talking about the Carl Linnaeus, and these are some points that I have shared with you, right, bache? So obviously from these points, this is what we can conclude, that he didn't pay attention on single-celled and multi-celled organisms. He didn't pay attention on the prokaryotic cells and the eukaryotic cells, right? Right. So in the Carl Linnaeus kingdoms, right, the bacteria, which is basically, you know, that prokaryotic cell, and protists, they all were placed together, isn't it? They all were placed together, isn't it? Right. So yes, he didn't even consider photosynthetic, non-photosynthetic, nothing at all. So obviously we need the better classification. So we have to go for the third kingdom classification, and it was given by Ernst Haeckel. Right. So we have to go for the next classification system, and that next classification system is three kingdom classification system, which is given by Ernst Haeckel. So bache, this is the thing, like first time Aristotle tried. Now Carl Linnaeus, he placed it in a better way. He used the word kingdom and all, and then from that two kingdom classification system, or whatever were the drawbacks now, right? So the next scientist, he studied that drawbacks, and he was like, okay, I'll do something better. Okay, I'll do something better. And that is why time to time we people used to tell you that kids start after mock test, start analyzing your paper, because from your mistakes you are going to learn a lot, right? So basically you can say that from the mistakes of the Carl Linnaeus, Ernst Haeckel, he learned many things, and he made what? He made the three kingdom classification system. So what he included? He included this thing, kingdom protesta, right? He included what? He included kingdom protesta. He included what, bache? He included kingdom protesta. Now in the kingdom protesta, what do we have? All the unicellular organisms. This is what they did, right? They put all the unicellular organisms all together. They were like, okay, this is the problem now. We are not paying attention to the unicellular organism. Let's put all the unicellular organisms all together. So fungi, protozoan, algae, bacteria, slime molds, all were there in kingdom protesta. Are you getting my point? All were there in kingdom protesta, right? So in kingdom protesta, unicellular organism plus the organism that lacks tissue differentiation, right? That lacks tissue differentiation, in which, you know, the tissue system is not there. So he kept everything there in the kingdom protesta. So obviously you know that we see the same thing in the case of fungus as well. So that is why he put everything in the kingdom protesta as well. So now you know that, okay, fine, if we are putting all the unicellular organisms in the kingdom protesta, now again you people can compare, right, bache? You guys can compare that, ma'am, what about prokaryotic and eukaryotic cell? What about prokaryotic and eukaryotic cell? Isn't it? There are some unicellular protozoans as well. There are some unicellular algae as well. They all are kept together. How is it possible? They have different properties, right? Are you getting it? So obviously it is not acceptable even. So we have to go for one better system of classification. So we were like, okay, this is the problem. Prokaryotic cell and eukaryotic cell is the problem. Okay, fine. We can introduce one more kingdom, and that will be kingdom Monera, and it was done by Copeland, right, bache? It was done by Copeland. So the name of these scientists, it is not given in NCERT, but it is important, my dear kids. Okay, it is not given in NCERT, but it is important, my dear kids. Okay, so Copeland kingdom, right? So Copeland, who gave it? So Copeland made the four kingdom classification system. So he introduced the kingdom Monera. Earlier the Monera was having the different name, some Mycota or something, but later on it was named as Monera. Is that clear? Is that clear? Right? right? So Copland named it as some, you know, mycota. This is the word that he used, but some other scientists, they renamed it as kingdom Monera. So you don't need that depth. So that is why I'm telling you, Copland, he gave the kingdom Monera. So now in the kingdom Monera, prokaryotic organisms were placed. In the kingdom Protista, all unicellular eukaryotic organisms were placed. Right. Kingdom Plantae and Animalia, you know that. Right, you know the basic points there. But again, there is one problem, and that biggest problem is kingdom Fungi. Right? Kingdom Fungi was the biggest problem. It is not even autotrophic. Right, it is heterotrophic, animal-like character, but it is not like animals. Some points are just like the plants. Right, so again confusion was there. They were not able to put the fungi properly. Are you getting my point? So finally, Mr. R.H. Whittaker came up with the 5 kingdom classification system. Right, so bache, remember it like a story. Trust me, remember it like a story. Even if you are not the dropper, if you are in class 11 and you don't like these words, ma'am, Monera, Protista, this and that, trust me, you will be habitual. Right, you will be habitual. Trust me. Right. Initially you are going to find it difficult, then it will be very easy for all of you. Very easy for all of you. Okay, so who gave that 5 kingdom classification system? Mr. R.H. Whittaker. R.H. Whittaker. He is the one who gave the 5 kingdom classification system. Is that clear, everyone? Yes. Is that clear, everyone? So obviously he was too smart. He was like, fungi is the problem. Don't worry about it. He was literally too smart. I consider him too smart. Seriously. He was like, fungi is the problem. Don't worry. So let's put it in that way. Kingdom Monera, all the unicellular prokaryotes. Kingdom Protista, all the unicellular eukaryotes. I will make kingdom Fungi. I will keep fungi in a different kingdom. Fungi is the problem, let's keep it in a different kingdom. And then there will be the kingdom Plantae and there will be the kingdom Animalia. So bache, these are the basic things. Right? So can we just recall it? So how do we start? We start with the Aristotle. Mr. Aristotle, the first scientific attempt to classify the organisms. In the case of plants, he was like on the basis of morphological characters, right? He looked at their size. He was like, if they are very small, herbaceous, herbs, shrubs and the trees. When it comes to the animals, it will be Anaima, Enaima, Anaima. A means without, right? Without red blood, without RBC, Enaima, with the red blood. Okay, that's how he categorized, and we know that it cannot be the appropriate way, right? It is not the appropriate way. It cannot be the appropriate way. Learn with Kazama. We will come to this as well. Don't worry about that. Okay, don't worry about that. Then Mr. Carl Linnaeus, right? Let's have a quick revision of these things, bache, because this can be asked as a question. Okay, then Mr. Carolus Linnaeus, he was like, okay, I will give it a try. So he gave that 2 kingdom classification system, plants and animals, on the basis of cell wall, on the basis of mobility, on the basis of response to the stimuli. That is how he categorized. But still it was not sufficient, because he didn't pay attention on the prokaryotes, on the eukaryotes. He didn't pay attention on the unicellular or on the multicellular. Then what we did, we called Mr. Ernst Haeckel. Right, now what Ernst Haeckel did, he was like, okay, let's make 3 kingdom classification system. So he introduced kingdom Protista. Right, he introduced kingdom Protista. So kingdom Protista came first. Right, kingdom Protista came first, and kingdom Protista, as per this Ernst Haeckel's kingdom classification system, kingdom Protista will include all the unicellular organisms. Right, all the unicellular organisms. Again, they didn't pay attention on the prokaryotes and the eukaryotes. Then what do we have? We have the 4 kingdom classification system. Copland, he introduced one more kingdom, that is Monera. So Monera include all the unicellular prokaryotes. Right, Monera include all the unicellular prokaryotes. Are you getting my point? All the unicellular prokaryotes. Then the finally 5 kingdom classification system, R.H. Whittaker. 5 kingdom classification system, R.H. Whittaker. First of all, tell me up to this part all clear. Then even I will tell you the drawbacks of R.H. Whittaker system as well. But first of all, tell me up to this part all clear. Yes, all clear. Yes, bacha, be quick. Be quick. Done? Okay. Sure? Yes, everyone? So please take a note of it. Please mention these points even in your notes. Okay, bache? So next is 5 kingdom classification system. We have to study about it in detail, and then we will talk about the 6 kingdom classification system. So the name of the scientist is very important. It is also a previous year question given in NCERT as well. So in 1969, right, an American taxonomist. So R.H. Whittaker was an American taxonomist. He classified organisms into 5 kingdoms. So what he considered? Can you tell me about that? Can you tell me what he considered? Anyone? What he considered? Anyone? Yes, bacha. So see, the session is detailed na? Session is detailed. है ना? We are not just teaching you from the advanced level, we are even including the basics as well. So what he considered, can you tell me? Can you tell me? Yes, what he considered, the very first thing that he considered is the cell structure, that what type of cell is there, right? Very first thing that he considered is the cell structure, that what type of cell is there. He paid attention on prokaryotes and the eukaryotes, right? He focused on these things, that cells are prokaryotic also, eukaryotic also, right? So the very first point is what? It is the cell structure. Second is thallus organization. Can you tell me what is the meaning of this thing, bache? Thallus organization. What is the meaning of this? Thallus organization. Thallus, right? Thallus word. We even use this word in plant kingdom. Right? So thallus is also, we are talking about the bodily organization. Like in the case of algae, we used to say na, it is a thalloid structure, not properly differentiated in true root, stem and leaves. This is how we discuss. So we are even going to focus on the cell structure. We will focus on the thallus organization. Here we will see that whether that organism is a unicellular organism or a multicellular organism. These are the points that are that we will consider, right? And then even they paid attention on the reproduction, right? Sexual, asexual, even it was considered. Sexual, asexual, right? Moreover, bache, nutrition, what type of nutrition is there? Even R.H. Whittaker, he even focused there, right? That what type of nutrition is there. So when it comes to the nutrition, you know that organisms, they can be autotrophic, means they can make their own food, and organisms can be heterotrophic, right? That is, they are dependent on other source. Done, bache? And finally, the most important thing is what? It is the phylogenetic relationship, right? It is the phylogenetic relationship. He even considered the phylogenetic relationship, means evolutionary relationship. He even considered how different organisms, they are related, right? Evolutionary relationship. Are you getting my point, bache? These points are important. MCQ can come from this part, right? In the NEET exam, they can ask you the question from this particular part. They can ask you that R.H. Whittaker, he considered which characters, right? Or they can give you the options out of the following, which characters were considered by R.H. Whittaker, which characters were not considered by R.H. Whittaker. So this is what you need to mark. This is what you people need to understand. So up to this part, all clear? Bache, evolutionary relationship, that how different organisms they are related, right? Do they have common ancestors or not? So that type of points were considered in the R.H. Whittaker classification system. So these are the, right, these are the features that he considered. Are you getting it? These are the features that he considered when it comes to the 5 kingdom classification system. Okay. when it comes to the 5 kingdom classification system. So please make the notes bache, it is important, it is literally very important to mark all these points, right? It is very important to mark all these points. Is that clear? Done? Done? Sure? Sure bache? Okay, so now here this table is important, right? After this we need to focus on this particular table. This table is important. You know that this table is from NCERT, so you have to remember this table. So this is the 5 kingdom classification system given by R.H. Whittaker. This is the 5 kingdom classification system given by R.H. Whittaker. So as I said, cell type. Now bache, all the 5 kingdoms are written here. All the 5 kingdoms are written here. Please take a note of it, because still many students, they make this mistake. They don't know about the difference in prokaryotes and eukaryotic organisms. Trust me. Trust me. You must be thinking, no ma'am, we are preparing for the NEET. We at least know the difference in prokaryotes and eukaryotes. No. No. Even if you are a serious NEET aspirant, right? Even if you are a serious NEET aspirant, you don't know the difference in prokaryotes and the eukaryotes, right? And you don't even know about these kingdoms and the classification system. So kindly focus here, right? So when it comes to the Monera, Monera is the only kingdom having prokaryotic cells. Rest all the 4 kingdoms are having eukaryotic cell. It's a previous year question, bache, right? It's a previous year question. Please mark it. Please mark it. Okay, that's important. That's literally important. So only kingdom Monera. It is the kingdom Monera only which is having the prokaryotic cell. Rest all the kingdoms are having eukaryotic cell. A very important difference. Is that clear? Now when it comes to the kingdom Monera is having unicellular prokaryotic cell. First of all, this is what you need to understand. It is having unicellular prokaryotic cell, and kingdom Protista is having, right? Kingdom Protista is having unicellular eukaryotes. Rest all will be. Exceptions are there, but still rest all are having multicellular organisms. Right, bache? So we need to understand this table. Okay, so please make the notes with it. So as per, now I am going to mention you the points only. As per this 5 kingdom classification system, when it comes to the Monera, in the Monera what do we have? We have the unicellular prokaryotic cells. And you know that the only member here is, although in detail also we will talk about this kingdom Monera, but still these are the things that you should know, right, from this table itself. From this table itself. So unicellular prokaryotes, and you know that only member here is bacteria. Only member here is bacteria. So these are the points from this classification. Now second, when it comes to the Protista, fungi, plantae, animalia, right? When it comes to the Protista, fungi, plantae, animalia, all 4, right? All 4, they contains eukaryotic cells. They contain eukaryotic cells. Now the third point is, when it comes to the kingdom Protista, right? So it will be having unicellular eukaryotes. It will be having unicellular eukaryotes. I'll quote one example, and many students, they don't even know that, right? Amoeba, paramecium. You know amoeba, right? It's a very famous organism. Paramecium, they all are the examples. They all are the unicellular Protista. They all are the unicellular eukaryotic cells, right? Right? We don't even know that. Sometimes, you know, I used to ask students, what is amoeba? Amoeba, they are like, it is unicellular. Yeah, you're right, it is unicellular. Then what else? It is placed in which kingdom? Arey, ma'am, it is Monera. It is bacteria. No, it's not like that. It's a eukaryotic organism. It's a unicellular eukaryotic organism having the proper nucleus, having the proper nuclear membrane. Okay. Okay. Right? Right? So, when you talk about the 4th point, that is your fungi, plantae, animalia, right? Your fungi, plantae, animalia, what are they, bache? They all are multicellular eukaryotic organisms. They all are what? They all are multicellular eukaryotic organisms. Is that clear? Yes, bache, is that clear? Right? So you know that here we have few exceptions, so you better note down these exceptions here only. You know that when you talk about the kingdom fungi, yeast, it's unicellular. Even if I go as per NCERT, chlorella is given in kingdom Plantae as per NCERT. So chlorella is also unicellular, right? Chlorella is also unicellular. Okay, bache? Okay, so these are few exceptions that you even need to remember as per that 5 kingdom classification system. And moreover, there are some other exceptions also that we discuss here. So up to this part all clear? Up to this part all clear? Now when we talk about this particular table, right, when we talk about this particular table, here in this table the cell wall, the nuclear membrane, body organization, mode of nutrition, everything is given. So bache, this question can come in different ways. Here they can, in the NEET examination, they can give you this table and they can, you know, put some blanks here. Ki what is A? Let's say here at the place of A, nothing is written, right? It's a blank. This fungi, this is a blank, this part is a blank, this part is a blank, like this. So they can ask you the question on the basis of that. So you should know everything given in NCERT, right? You should know the meaning of each and every line there. So if it is, now let's relate it. So if the cell type, right, if the cell type is Protista here, if Monera is prokaryote, Protista, fungi, Plantae, Animalia, eukaryotes, right? They are the eukaryotes. So obviously if it is a Protista, the nuclear membrane will be absent. These 2 points are interrelated, right? Right, these 2 points are interrelated. So if the, right, bache, if it is a Protestin, so of course nuclear membrane will be absent there, and rest all are eukaryotes. So of course in their case nuclear membrane is present, isn't it? In their case nuclear membrane is present. Now I told you, just now I told you that even your kingdom Monera is unicellular, even your kingdom Protista is unicellular, rest all are multicellular. So here in kingdom fungi they are multicellular, but tissue differentiation is not proper. Loose tissue is there, right Kriti? Loose tissue is there. When it is Plantae, here you will be having the tissue and organ, and in the Animalia, the cellular level, the tissue level, the organ system level will be there, right? Multicellular structures will be there, just a cellular structure. I'll quote one example. I hope you all remember this, right? So you know na, Porifera. In kingdom Animalia we have the phylum Porifera. So phylum Porifera is just a multicellular structure. Here we are having the cellular organization. What do we have here? We have the cellular organization. Is that clear, bache? Is that clear? Right, right. So this is what you will get. Okay, so now when it comes to the mode of nutrition, right? So mode of nutrition in Monera, it can be autotrophic, right? It can be heterotrophic. We will discuss it in detail. Individually we will start kingdom Monera, Protista, we will discuss it in detail. Now when it is kingdom Protista, autotrophic, again heterotrophic. In the fungi, it is going to be heterotrophic only. It cannot be autotrophic. So this can be asked as a statement based question. Fungus, are they autotrophs or heterotrophs? Always heterotrophs, and moreover they are having saprophytic nutrition, that is they feed from the dead and decaying organic matter, right? They feed from the dead and decaying organic matter. Saprophytic it is. Saprophytic it is, and it can be parasitic as well. Parasites they are, right? They depend upon a host. They will take their nutrition from them. Okay, and then Plantae, always autotrophic. Animals, always heterotrophic, right? Mode of nutrition we will discuss it in detail. So if there is any doubt from this particular part, do let me know. Even if there is a single doubt from this particular part, do let me know, kids. Do let me know. Yes, do you have? Kids, do you have? Sure? Sure? Are you sure? Okay. So now come to the next system of classification, that is six kingdom classification system. But in your syllabus you just have 5 kingdom classification system, so we are going to revise that only. So we have one more system, that is 6 kingdom classification system. Right, what do we have, bache? We have 6 kingdom classification system. So this system of classification was given by Carl Woese. Carl Woese. Right, we started with the 2 kingdom, Carl Linnaeus. 6 kingdom, Carl Woese. We will discuss it, Ashika, one by one we will discuss it. Okay, Soumyadeep, it's Carl Woese. Now, when it is the 6 kingdom classification system, I don't know what is. Okay, so when it is 6 kingdom classification system, as I said, it was given by Carl Woese. So Carl Woese, what he did, he divided bacteria basically. What he did, he divided bacteria, or you can say that he classified bacteria. Right, he classified bacteria on the basis of presence or you can say that absence of peptidoglycans, right, and branched lipids. I will explain it in detail, but basically what Carl Woese did, he divided bacteria. He divided bacteria on the basis of presence or absence of peptidoglycan and the branched lipid. Right, branched lipids. So he introduced one more kingdom, that is archaebacteria. He divided bacteria basically. So he introduced one more kingdom, that is archaebacteria. Right, he introduced one more kingdom, that is archaebacteria. Are you getting it? Are you getting it? So here in the case of Carl Woese, right, right, as per him, when you talk about the bacteria, there are 2 things. One is archaebacteria, ancient bacteria, primitive bacteria, which can survive in extreme conditions. Right, which can survive in extreme condition. And another is the eubacteria, true bacteria. Okay, so he compared archaebacteria and the eubacteria. What he did, he even compared archaebacteria and eubacteria. So when it comes to the archaebacteria, A for, this is how you have to remember, A for ancient. Now in the case of archaebacteria, cell wall, right, cell wall does not contain, it does not contain peptidoglycans. Are you getting it? Peptidoglycans are absent here. It does not contain peptidoglycans, and you can say that murines or muramic acid. Fine. And now when it is the eubacteria, U means true bacteria. So it will be having peptidoglycan. We will discuss the structure in detail, bache. Let me start Kingdom Monera. We will discuss everything in detail, but as of now just focus here, right? So in true bacteria, peptidoglycan, it is present in cell wall. Fine, it is present in cell wall. Are you getting it? Now the another point here is here the cell membrane, it is not bilayered, it is single layered. Cell membrane is single layered here. It was not bilayered. It was single layered. And moreover fatty acids, they were branched. So basically you can say that branched lipids were there. Right, branched lipids were there. Is that clear? This is important, bache, please note down. This is important, kindly note down. So what is the point here? In the case of archaebacteria, the cell wall is not having the peptidoglycan, right, and branched chain lipids were there, right? Branched chain lipids were there. Lipids, they are made up of fatty acid, glycerol. This is what we know now. So basically the lipids, they were what? They were branched. Okay, they were branched. So now the layer is monolayer, the lipids, they were branched. So obviously fluidity will be less here in archaebacteria. Fluidity will be less here in archaebacteria. Yes or no? Yes or no? Tell me quickly. We are talking about the archaebacteria. I am telling you A for ancient, primitive it is. So when you talk about the cell wall, it does not contain peptidoglycan and muramic acid, right? And when you talk about the cell membrane, it is single layered, it is not phospholipid bilayer, and moreover the lipids, they are branched, and branched lipid means less fluidity. Less fluidity means the membrane is going to be solid. The membrane is going to be solid means it can survive in the extreme conditions, it can survive in the harsh conditions. Getting it? It can survive in the harsh conditions, it can survive there. And now when it comes to the eubacteria, peptidoglycans are present, right? Phospholipid bilayer is there, right? Phospholipid bilayer is there. Fine, fine. So on the basis of these points, so Carl he classified the bacteria. He classified the bacteria, so he introduced one extra kingdom basically. So he introduced kingdom archaebacteria. Rest everything is same bache. He introduced kingdom archaebacteria. This is extra that he introduced, right? So basically he bifurcated. This is what I can say, that it is a kind of bifurcation of kingdom Monera, right? In kingdom Monera, all the bacterias were there. All the bacterias were there. So he was like, no, I will break kingdom Monera. I will divide it into two. So archaebacteria, another kingdom was eubacteria. Then rest all four are same. Like your kingdom Protista, kingdom Fungi, kingdom Plantae, and even the kingdom Animalia. So it was all same. It was all same. So kingdom archaebacteria, this is what he introduced. Okay, so eubacteria, Protista, Fungi, Plantae, and Animalia, right? Animalia. So this is about the 6 kingdom classification system Carl was. But here there is one more point, and that is about the domains, right? There is one more point, and that is about the domains. So basically Mr. Carl was bache, right, he introduced the word domain. He was the one, and it is given in NCERT. I'll tell you where. He introduced one new category, and that category was domain. What was it? It was domain, right? He introduced the new category, that was domain, and as per him, domain is the highest category. As per this system of classification, it is the highest category. Fine, it is the highest category. Okay, the domain. So 3 domains were introduced by him. 3 domains. So one was, yes bache, one was archae, another was bacteria, and the last was eukarya. Is that clear? So one was archae, another was bacteria, another was eukarya. So you know that here in archae, the kingdom will be archaebacteria. The kingdom will be archaebacteria in this domain, right? So domain will be the highest here. Here in bacteria, you will be the eubacteria as the kingdom, and here in eukarya, we will be having the kingdom Protista, we will be having the kingdom Fungi, the kingdom Plantae, and the kingdom Animalia. Okay, and the kingdom Animalia is there. Is that clear? Yes? Sure? So this is about the basic introduction of these systems of classification. Okay, this is about the basic introduction of these systems of classification. Now we have to focus few points from the NCERT, and then after that kids, after that we will be starting the Kingdom Monera. So up to this part, if there is any doubt, do let me know. That yes ma'am, we are having the doubts. Yes, do you have any? But I will share the PDF notes for sure this time. Pakka I will share it. Progenote. Nice bache Vishnu. Nice, nice, nice. Although it is not given in NCERT, but still. Okay. So, as per Carl was, right, Vishnu asked one question. I told you that Carl was, he gave the 6 kingdom classification system. And I told you that he basically studied the bacteria and he kept it in different kingdoms. As per Carl was, they all originated from the same ancestor, and that is progenote. Right Vishnu? So progenote is nothing. Progenote is basically the ancestor here. Right? Progenote is nothing. Progenote is basically the ancestor here. Is that clear? Is that clear, Vishnu? Yes, bache. Is that clear? Sure? Sure. Right? So progenote is basically the common ancestor for all the domains. It is basically the common ancestor for all the domains. Right? All the domains, they originated from the common ancestor. So let me write down this. Progenote is the common ancestor of all 3 domains. Anything else, bache? Any other doubt? Any other doubt? Can we go ahead? Yes, can we go ahead? Very good. So now we will start from NCERT. Okay, see, NCERT reading is important. So we are done with the introduction part. So now let's read the points from NCERT. So meanwhile your NCERT will also be clear, and you guys can make the short notes with the class itself. Okay, so whenever you attend the class, please keep a notebook with you, bache, and just take a note of it. And moreover, one more thing is there, bache. See, on Sunday in Avengers batch, there is one mock test, right? On Sunday in Avengers batch, there is one mock test. That mock test is for the students who are the part of the Avengers batch. But on Monday, I am going to explain all the questions of this mock test in a special class. So there will be a special class. That special class is at 4 p.m. Right, so we will do the question practice of chapter cell cycle and cell division. Right, so it is basically a question practice session of cell cycle and cell division, and this special class, it is free of cost, so anyone can join it. Right, it is free. You just need to download the Unacademy app, or on Google itself you can directly log in. Right, you guys can join it. Fine. So on Monday, special classes there at 4 p.m. free of cost. Right, it is for you, anyone can join it, bache. You just need to download the Unacademy app, and we will do the question practice of chapter cell cycle and cell division. So I will take your doubts as well. Fine, I will take your doubts as well. Okay, so see, as per NCERT, in Linnaeus time, a 2-kingdom system of classification with Plantae and Animalia was developed, and it had the plants and animals respectively. Any doubt here? Yes, any doubt here? Is that clear? Up to this part, is that clear? Now the next thing, hidden, right, the loopholes. Just focus on the loopholes as well. So hidden, distinguish prokaryotes, eukaryotes, unicellular, multicellular, and photosynthetic and non-photosynthetic organisms. Clear, bache? So in your notes, this is what you need to add. In your notes, this is what you need to add. Afia, ma'am, progenote is having all the characters, but it is the ancestor. From that ancestor, all the 3 domains, right, they form. So basically you can say that in different directions it evolved. right? Done. So this is what you need to include in your notes. You have to write down drawbacks of two kingdom classification system. These are the points that you need to mention there. Okay, bache? Okay, so classification of organism, as I said, into plants and animals, it is very easy to understand, but we know that we cannot put all the animals there, right, bache? Right, bache? So here, besides gross morphology, besides means in addition to gross, gross means superficial, right? What is the meaning of gross? Gross means superficial. So in addition to that superficial morphology, we also should know, right? We should know about the cell structure. We should know about the nature of the cell wall. We should know about the mode of nutrition, the habitat, the methods of reproduction, the evolutionary relationships, etc. Right? So here we are talking about what? We are talking about the 5 kingdom classification system. What are we discussing here? We are discussing the 5 kingdom classification system. Isn't it? Isn't it? Isn't it? So question can come from this part as well. They can ask you that which characters are considered for the 5 kingdom classification system. So in addition to the morphology, the cell structure, the cell wall, mode of nutrition, the reproduction, the phylogenetic relationship, mode of habitat, everything is considered. Okay, bache? Okay, bache? Done? Done, done, done? Okay, so R.H. Whitaker in 1969, so he was an American taxonomist. He gave the 5 kingdom classification system. Okay, so main criteria that he used was, as I said, cell structure, body organization, also known as thallus organization, mode of nutrition, reproduction, phylogenetic relationship. So this is important, and it is also a previous year question, bache, killer science, bache diya, which is Surjaya Pragati, it is also a question, right? So in the question it is given that R.H. Whitaker included dash in the classification. During its classification he considered dash. So phylogenetic relationship was given and some other characters were also there, but many students, they didn't mark the phylogenetic relationship. Why is it so? Because they do not read the NCERT properly. So see, if you will just focus on this particular table, right, if you will just focus on this particular table, here in this particular table, right, it is the 5 kingdom classification system, but they didn't mention the phylogenetic relationship, isn't it? Phylogenetic relationship is not mentioned here. So that is why we need to read the NCERT properly, right? We need to read the NCERT properly so that we can give answer of each and every question. So bache, what are the minimum marks that you want to score in the biology? Tell me. First of all, tell me this. What are the minimum number of marks that you want to score in biology? I'm not asking maximum, minimum. Minimum. You don't know the maximum number, right? But you should know your minimum. That okay, below 330 I will not go. For sure, I have to score 330 in the biology. Right? I have to sure, at any cost, I have to score 360, 330 marks in biology. So basically your minimum is 340. I'm not saying 320 plus 330, minimum. For some students it can be 310, 320, 330. So if you want to score 300 plus marks, right, if you people want to score 300 plus marks, then you need to read each and every line of NCERT. You should understand each and every line, and moreover the tables given, the diagrams given, everything is important when it comes to read the biology, NCERT biology. Fine, so discuss, so read it properly, and with classes please make the notes. Fine. So now come to the next part. So classification system for the living organism, you know that it has undergone several changes over the time, right bache? Right bache? So you know that, some in earlier classifications, some animals, some you can say that animal cells, they were placed in the kingdom Plantae, then they kept in different, like basically you know that with the time, right, with advancement, with better instruments, right, we are able to study the structures more clearly, and then on the basis of that we can put the organisms into different kingdoms properly. So yes, even in future there are chances that we can change this classification system with the better clarity, with the better understanding. Okay, done. Done bachche. So now after this paragraph is also very important. So see here, the 3 domain system is given. So many times students used to ask me that 6 kingdom classification is not given in NCERT array, it is given 3 domain system. So it was given by Carl Voss. So even in NCERT you guys can write it down. Fine bachche, you people can write it down. Now this paragraph is also very important. So let us look at this 5 kingdom classification to understand the issues and consideration that influence the classification system. So when you talk about the earlier bachche, when you talk about the earlier classification systems, bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms, all were there in plants. Right? If I talk about, right? If I talk about what? If I talk about the earlier system, so as per that, in kingdom Plantae, bacteria, blue-green algae, fungus, mosses, ferns, gymnosperms and angiosperms, all were kept there in the kingdom Plantae. This is how you need to make the notes as well. But now you know that these two, they are the bacteria. Now they are the part of Monera. Fungus is a different kingdom now. Mosses, ferns, gymnosperm, angiosperm, they all are the parts of, they all are present in the kingdom Plantae itself. Right, they all are present in the kingdom Plantae itself. So here you can see the character that unified this whole kingdom was that all organisms, they include the cell wall. Right, so on what basis? Bacteria, blue-green algae, fungi, mosses, they all were kept in kingdom Plantae because they all were having the cell wall. So guys, this is how you have to make the notes. So see, this is a paragraph, you have to just write the word plants, right? And here you can even give a title, even give the heading, earlier classification. As per earlier classification. So all of them were placed in kingdom Plantae, and what was the basis of that? The cell wall, the presence of cell wall. Is that clear bachche? The presence of cell wall. Is that clear? Yes everyone. Yes everyone. So this placed together groups which widely differed in other characteristics. Right, so it brought together the prokaryotic bacteria, the cyanobacteria with other groups which were eukaryotic. So obviously this is what we can depict from this now, that these prokaryotes, they were placed together with the eukaryotes. These prokaryotes, they were placed together with the eukaryotes. Right bachche, and it also grouped the unicellular organism and multicellular. I have already given you the example of Chlamydomonas and Spirogyra. So Chlamydomonas being unicellular, right, being unicellular, Spirogyra being multicellular, being photosynthetic also, they were placed under algae. Right, they were placed together under algae. So they are saying, let us look at the 5 kingdom classification to understand the issues and consideration that influence the classification system. So here you people can see, okay, so Chlamydomonas, Spirogyra, they all were kept under algae. Right, one is a unicellular, another is multicellular. So classification, it didn't differentiate even the heterotrophic group fungi and autotrophic green plants. Right, and not even the difference in their cell wall composition. You know that fungus is having the chitin, right, in their cell wall, and when it comes to the green plants, the cellulosic cell wall is there. Right, it is also a PYQ. The fungus cell wall is having chitin. Right, fungus cell wall is having what? Chitin. So that chitin is what? It is a homopolymer of N-acetyl glucosamine. Okay, chitin is what? It is a homopolymer of N-acetyl glucosamine. N-acetyl glucosamine. Is that clear? Is that clear? And when it comes to the plant, cellulose is there, and you know that cellulose is what? It is the polysaccharide, right? It is a homopolysaccharide. It is also a homopolysaccharide. Fine? Cellulose and chitin both are homopolysaccharides. Right? Now bachche, I know here in this line there is a confusion, like Chlamydomonas and Spirogyra, they were placed together under kingdom planty. I know this line is little confusing, Afia. This line is little confusing for all of you. See, actually what is the scenario? No doubt we discuss these chlamydomonas and all in kingdom algae, right? We talk about the chlorella, we talk about the chlamydomonas in kingdom algae, but basically it is not like that. These are the unicellular eukaryotic cell. Actually they are the part of kingdom protista. Actually they are the part of kingdom protista, but it is much more related to algae, so we study it there. Okay, we study it there, right bache Afya? Okay, okay. So when such characters were considered, the fungi were placed in a different or in a separate group. Now you got it. So all prokaryotic organisms were grouped in kingdom Monera, unicellular grouped in kingdom Protista. Kingdom Protista brought together chlamydomonas and chlorella, which were earlier placed in algae within plants, and both having cell walls, right? So this point is very important, and I know many times students, they get confused here. Like Afya asked that, ma'am, here it is written that chlamydomonas and spirogyra, they are the part of kingdom plantae only. No, chlamydomonas is the part of kingdom protista, right? It is the part of kingdom protista. It is unicellular, having the cell wall, right? So, but where do we study it? Where do we study it? We study it in kingdom plantae, right? Because so many chlamydomonas together will form that wall box colony and all, na? That is why. And same is the question here. Same is the question here. So as per this 5 kingdom classification system, chlamydomonas, chlorella, which were initially placed in algae, right, but now they came closer, brought closer in kingdom protista. So you got it? We study it there, but they are the part of kingdom protista. Is that clear? Is that clear? That is why, bache, I added this NCERT snip to make everything crystal clear to you all. Fine? Yes, bacho, do let me know in the chat section, guys. Do let me know in the chat section. So chlamydomonas and chlorella in the protista, and paramecium and amoeba, which were earlier there in the animal kingdom. So because of this 5 kingdom classification system, we kept it in kingdom protista, right? We kept it in kingdom protista. You got it? So earlier amoeba and paramecium, because they lack cell wall, so they were kept in animal kingdom. But because of this 5 kingdom classification system, where we have studied the cell structure, the body organization, right, reproduction, nutrition, everything we have studied there, so we kept it where? In the kingdom protista. Okay, bache? Okay, done. So it happened because the criteria of classification changed. So as I said, it can occur in the near future as well, bache, right? So over time an attempt has been made to evolve a classification system which is going to reflect morphological, means outer forms, physiological, internal functioning, reproductive similarities, but it also include phylogenetic relationship, that is evolutionary relationship, that how 2 organisms are related together. Okay, that how 2 organisms are related together. Is that clear? Is that clear? Right. So now we have to start the kingdom Monera, but before that, if there is any doubt, do let me know. But before that, if there is any doubt, do let me know. Protista unicellular, Lakshmi, seriously, kingdom protista, it is itself a kingdom. It is having all the unicellular eukaryotes. Bache, on Monday in special class, cell cycle and cell division, this chapter, the question of this particular chapter will be discussed. Okay, so clear? Yes, so can we start kingdom Monera? Yes, bache, can we start kingdom Monera? So one more thing, your viruses, your lichens, viroids, prions, they all are exempted from your 5 kingdom classification system. Viruses are not the part of 5 kingdom classification system. Neither your viroids, prions, right, they are not the part. Even lichens are not the part. What is the reason behind? See, when you talk about the virus, you know that it is a connecting link in between living and non-living organisms, right? It is what? It is a connecting link in between living and the non-living organisms, isn't it, bache? Isn't it? What are we talking? We are talking about the virus, right? We are talking about the virus. So outside the body it is just a inert protein. Inside the body it is living. It will use the host machinery. Lichen is having algae and fungi both in the symbiotic relationship. Viroids even smaller than viruses. Infectious RNAs these are. So they are not kept in any of the kingdom. So we will talk about the we will talk about the viruses, the viroids also, but after finishing this Monera, Protista, fungi. Okay, after finishing this Monera, Protista and fungi. So any doubt? Any doubt? Bache, Anul is asking, ma'am, in saprophytic bacteria and saprophytic fungi are same. Bache, one is bacteria, one is fungi, but the word saprophytic is same. So saprophytic is organism which feeds on the dead and decaying organic matter. Which feeds on the dead and decaying organic matter. This is the key word here. Okay. Okay, so now can we proceed? So what we have to discuss? We have to discuss the kingdom Monera. So you know that kingdom Monera was introduced when there was the four kingdom classification system was there, right? When four kingdom classification system was there. Is that clear? Is that clear? So when it is kingdom Monera, bacteria are the sole members, right? Bacteria are the sole member. Isn't it? Bacteria are the sole member of kingdom Monera. Is that clear? Is that clear? Yes, bache. So what will we have here? We will be having a prokaryotic cell. So here in kingdom Monera, we are going to study the Archaebacteria, the Eubacteria, the Mycoplasma, right, the Mycoplasma and Cyanobacteria, which is also known as photosynthetic bacteria or blue-green algae, right? Even actinomycetes are also there. Right, even actinomycetes are also there when we talk about what? When we talk about the kingdom Monera. So it is the bacteria which is the sole member. It is the bacteria which is the only member. Okay, which is the only member. Is that clear? Is that clear? Yes or no? Yes or no? So bacteria, bache, bacteria, you know that they are present everywhere, है ना? Bacteria, it is present everywhere, isn't it? It is present everywhere, and you know that it is microscopic. It is microscopic, and bache, we are discussing na, more than 1.5 million species are there on planet Earth. So in that list, we are not including the bacteria. Right, in that list we are not including the bacteria. What is the reason there? Why are we not including the bacteria there? But because we do not have bache such resources that we can study or we can grow all type of bacteria. Even if we start classifying the bacteria now, even in the bacteria of same species, you can say that in different strains you will see diversity, you will see variety, even in their cell wall, even in their structure, right? Even in their cell wall, even in their structure. Okay, bache? Okay, so now let's talk about the kingdom Monera in detail, right? So these points are very simple, that they will be unicellular, prokaryotes, no membrane bound organelles are there, genetic material is nucleoid, genophore, right? 70S ribosome is there. These are the general characters. So now before proceeding, right, we are going to discuss the structure of bacteria. Okay, bache, right? So before starting all that different types of bacteria, let's talk about the structures of bacteria, or let's talk about the structure of a prokaryotic cell, right? So bache, what do you know about a prokaryotic cell? Yes, what do you know about a prokaryotic cell? Anything that you know about the prokaryotic cell? Afia, let's discuss it, have patience. Okay, let's discuss it one by one. First of all, let's talk about the structure now. So let's discuss the structure first, then we will talk about the shape, the type of flagella, right, the type of bacteria. But first of all, let's finish it. Okay, let's finish it. So when it comes to the structure of bacteria, the very first thing that we need to know is the outer coverings, right? The very first thing that we need to know is what? It is the outer coverings. So bache, there are 3 protective outer coverings. What do we have in a bacteria? We have 3 protective outer coverings, right? So we are discussing the structure of bacteria and we are starting with the outer coverings. Outer coverings. So what do we have? We have 3 protective coats. 3 protective coats. So if I tell you about the sequence from outer side to inner side, right? This is the sequence from outer side to inner side. So outermost will be your cell coat, also known as glycocalyx, right? Then comes your cell wall and innermost is your cell membrane. Are you getting it? Innermost is your cell membrane. Okay? Innermost is what? Innermost is your cell membrane. Clear, bache? Clear? So what are we discussing? We are starting with the yes, bache, we are starting with the outer coverings first. So 3 protective coats are there from outer side to inner side, right? Like from outside to inside, if you check the structure of bacteria. So outer side, as I said, it is the cell coat, also known as glycocalyx. Also known as glycocalyx. Then comes the cell wall. Cell coat and cell wall is different. And then innermost is the cell membrane. So you need to know about it. Fine? You need to know about it. Clear, bache? Clear? Yes, is that clear? Sure? Okay, so let's start with the cell coat. So let's start with the cell coat first. So bache, when it comes to the cell coat, we also call it as glycocalyx. Right, it is also known as glycocalyx. Actually, there are 2 things when it comes to the cell coat. One is the capsule, another is the slime layer. Right, one is the capsule, another is the slime layer. Bache, capsule is tough, made up of polysaccharides, of course, but it is tough. Tough means it is solid, right? Tough means it is solid. You can relate it with the Griffith's experiment, right? In class 12th, there is Griffith's experiment. In class 12th, there is Griffith's experiment. In that Griffith's experiment, we have the smooth S-shape, S bacteria, right? Smooth bacteria, it forms the smooth colonies. It is virulent. It used to cause the, right, pneumonia. Another is what? Another is your R-shaped bacteria, rough strain. It is not forming the colonies. It is non-virulent. We discuss the words like this, isn't it? Isn't it? So when it comes to the capsule, bache, when it comes to the capsule, so it is the tough, right? It is what? It is the tough outer layer. It is made up of polysaccharide. It is compact. And when it is the slime layer, bache, right? When it is the slime layer, it is what? It is jelly-like, right? What is it? It is jelly-like. It is a loose slime layer. Okay, it is a loose, it is the slime layer. So this is what you need to understand first of all. So if it is a loose sheath, it's the slime layer. If it is thick and tough, it is the capsule. Okay, it is the capsule. Okay, so glycocalyx will include this. Done, bache? Done? Okay, so ultimately this glycocalyx is one which can hold water, right? So anything which can hold water, we use the word mucilaginous for that, isn't it? Isn't it? Isn't it? Like here, if I talk about the glycocalyx, so what we used to call it, we used to say that this is outermost mucilaginous covering. So mucilaginous means it can even hold the water, right? It can even hold the water, and it is also made up of what? It is also made up of mucopolysaccharides, right? It is made up of mucopolysaccharides. Is that clear? So bache, when it is the capsule, the tough thick layer made up of polysaccharide, so it protects bacteria. Okay, it protects bacteria from desiccation, okay, from antibiotics. Are you getting it? One is thick, another is the slime layer, so obviously it is as per their adaptations. Okay, so one is going to protect the bacteria from the desiccation, from the antibiotics, right, from the desiccation, from the antibiotics, and even from the viral attack, and when you talk about this jelly-like, so obviously it is going to do what? It will hold the water, right? It will hold the water. Slime layer it is, mucilaginous it is. Okay bache, so it will protect the cell from loss of water and nutrients. Okay, it protects from loss of water and nutrients. So this is the outermost covering. So bacteria can have capsule, it can have the slime layer. So is there any doubt? Is there any doubt? Yes, is there any doubt bache? Tell me quickly. So cell coat, also known as glycocalyx, outermost mucilaginous covering made up of mucopolysaccharides. It can be with or without proteins, right? These polysaccharides can have, like basically outer composition can have proteins or cannot have, means with or without proteins. Okay, so two things are there, capsule, slime layer, tough, made up of polysaccharides, jelly-like. Clear? So first covering done bache, So next is what? Next is the cell wall. Okay, next covering is what? It is the cell wall. So first of all, let's talk about the cell wall. So bache, when it comes to the cell wall, in the case of bacteria, the cell wall is made up of peptidoglycans, right? It is made up of peptidoglycans, no doubt. In your archaebacteria, peptidoglycans are not there, but mostly your bacteria is made up of what? It is made up of peptidoglycans. So peptidoglycans, also known as murines, right? They are basically the mucopolysaccharides. They are also the mucopolysaccharide. Okay, mucopolysaccharides. So what are we discussing? Ma'am, cell coat not present. Bache, Gibran, the cell coat is there in the bacteria. Bacteria is having these three outer coverings. Plants are just having the cell wall. Animals are just having the cell membrane like this. Okay, so when you talk about the cell wall made up of peptidoglycans, also known as murines, basically they are mucopolysaccharides, right? So which peptidoglycans are there in the case of, it's an important question bache, which peptidoglycan is there in the case of bacteria? It is your NAM, it is your NAG. It is your NAM, it is your NAG. So bacteria cell wall is not containing the cellulose. Cellulose is the part of plant cell wall. Chitin is the part of fungal cell wall. When it comes to the cell wall of the prokaryotic cell wall of the bacteria, then what do we have? The peptidoglycans. What is the name here? The name is NAM, NAG. N stands for N-acetyl-muramic acid. N-acetyl-muramic acid, right? And G stands for N-acetyl-glucosamine. Okay, G stands for N-acetyl-glucosamine. So these structures, they are kind of monomers. They will together form these peptidoglycans. Okay, so along with these peptidoglycans, some short peptides are also present there, right? It is not just these peptidoglycans are present. No, along with that, some peptides, some short peptides are also there, right? Some short peptides are also there. Are you getting it? So cell wall is made up of peptidoglycans, purines and polysaccharides and some short peptide. Done, bache? They are also there in the cell wall of bacteria. Now, there is one question. You know that in the case of bacteria, we know that we have the antibiotics. We have the antibiotics that can destroy that bacteria, right? That can destroy that bacteria. Even whenever we feel sick, if it is the bacterial infection, doctor is going to give us the antibiotics. Doctor will be like, have this for 3 days or 4 days. So it is going to destroy your bacteria. Basically, there are different ways to kill a bacteria, and out of that, one is to destroy the cell wall. Right, out of that, one is what? To destroy the cell wall. Some antibiotics, they do not let the cell wall to grow, right? And some antibiotics, they hydrolyze the structure of cell wall, right? So bacteria will not be able to survive. So now this is your homework. I want to see the answer in the comment section, that which antibiotics they act on cell wall. Okay, which antibiotics they act on cell wall. This is what you people need to tell. Okay, this is what you need to tell. Is that clear? Is that clear? Done? Done. Okay, fine. Sure. Are you sure? Okay, so this is about the cell wall. So some short peptides are also there in the cell wall. They are cross-linked. Okay, they are cross-linked. So now you know that on the basis of cell wall, you have two types of bacteria. Can you tell me? Can you tell me the name of that bacteria? On the basis of cell wall also, we have two types of bacteria. Can you just name it? Which bacteria do we have on the basis of cell wall? Which two types do we consider when it comes to the cell wall? Yes, bacho, it's a very common question. It's a very common question. No? Very good, very good. So bacteria, right? Bacteria on the basis of bacterial cell wall, you know that one is gram positive bacteria, right? One is gram positive bacteria and another one is what? It is the gram negative bacteria. One is gram positive bacteria, another is gram negative bacteria. So now bache, what is the story here? See, gram positive, gram negative. Basically, there is one scientist whose name is Kristen Gram, right? One scientist was there whose name was Kristen Gram. He made one stain. Stain means color, of course, right? Stain means color, of course. Stain, the color which is going to color that particular structure. It can be cell wall, it can be any organelle. Stain is related to the color. So Kristen Gram, he made one stain. The name of the stain was basically the crystal violet, right? It was the crystal violet, right? So any bacteria which is giving color with this stain, right? Bacteria which is giving color with the stain, that bacteria was gram positive. That bacteria was gram positive, isn't it? Isn't it? If a bacteria is giving color with the crystal violet stain or basically with the gram stain, this is the gram stain, it is having the crystal violet, I'll tell you about it, right? So Kristen Gram, he made a stain which is known as gram stain. So any bacteria which is giving color with that particular stain, that bacteria is the gram positive stain. And if not giving color with gram stain, okay, if it is not giving color with gram stain, then the bacteria will be gram negative. Bacteria will be gram negative. Now, on what basis we can decide it? It depends upon the bacterial cell wall. So, ma'am, take a session, but this is for the droppers, na? Okay, so gram positive, gram negative, gram positive, gram negative. So gram positive, gram negative, gram positive, gram negative. So it depends upon the difference of the cell wall. Now, if I talk about the gram positive bacteria, so you know that innermost covering is going to be the cell membrane. Yes, everyone? Just tell me, innermost covering is going to be the cell membrane, yes or no? Bache, please quickly type it in the chat section. I told you the sessions will go from basic to advanced level, right bache? Right, we have promised you now that sessions will go as per basic, from basic level to the advanced level. So I am explaining everything here. So please, 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 all of you, please answer it quickly. You know that cell membrane is the innermost covering. Cell membrane is the innermost covering. Yes or no? Yes or no? Right now we are talking about the cell, right? We are talking about the cell wall. So basically on the basis of that cell wall, right, we know that bacteria, we are even going to categorize bacteria into two types, gram positive, gram negative. So when it is the gram positive bacteria, when it will give color with the gram stain. I will tell you the procedure also, but as of now just listen to me, right? If it is not giving the color with the gram stain, it is the gram negative bacteria. So all that things, it depends upon what? It depends upon the structure basically here. So when you talk about the gram positive bacteria, here the obviously innermost is your cell membrane, and outermost, this outermost covering is your cell wall, right? Outermost is your cell wall. And here, when you talk about gram negative, see, just look at the structure of gram positive and gram negative bacteria. Just look at the structure of gram positive and gram negative bacteria. Just have a look here. So here you guys can see, this portion is what? It is the cell membrane, right? It is the cell membrane. Now when you look at gram positive bacteria, here in gram positive bacteria, cell wall is very thick, right? Here in gram positive bacteria, cell wall is thick, is very thick, isn't it? It is very thick. This layer which is made up of peptidoglycan, it is very thick. And here if you focus here, you know that the cell wall is not so thick, right? Cell wall is not so thick. And here in the gram negative bacteria, there is one more lipid outer envelope. It looks like the lipid now, it looks like the lipid. So by just looking at this diagram, you can say so. So outermost, this is the lipid envelope, right? This is the outermost lipid envelope. So just compare these two structures here. Just compare these two structures here. In gram negative bacteria, you are having the lipid cell membrane. The cell wall made up of peptidoglycan is very thin, it is not so thick, and moreover in addition to that, there is one outer envelope, outer covering, which is also made up of the lipids. This part clear? Which is also made up of lipids. Now come here to the gram positive bacteria, right? Here we are having the inner cell membrane, here we are having the cell wall. Cell wall is too thick, you know that, it is made up of peptidoglycans. It is made up of peptidoglycans, it is too thick, and moreover this cell wall is also having the teichoic acid. It is also having the teichoic acid, right? It is also having what? It is also having the teichoic acid. This teichoic acid is absent here in gram negative. Now because of the thick structure, right, this structure is going to hold the gram stain, right? It is going to hold the gram stain. Are you getting my point? It is going to hold the gram stain, it will not let it come out, and here the gram stain will come out. Now I will tell you the procedure, but first of all, it is teichoic, T-E-I-C-H-O-I-C. But first of all, tell me, up to this part all clear, bache? Is there any doubt? Is there any doubt? No? So now I'll tell you the procedure, right? I'll tell you the procedure of this gram stain. The procedure here is, come in and stain, right? This is the trick. Come in and stain. Come in and stain. Whenever we stain something, right? Whenever we stain something, whenever we color something, basically what you are going to do, there will be a tray, you are going to put the cells there, there will be the, you know, there will be some steps that you need to follow, isn't it? There will be some steps that you need to follow, isn't it? Yes or no? Yes or no? Like here, if I talk about the bacteria, so first of all, what will I do? I will take the culture of bacteria. I will take the culture of bacteria, right? I am not going to teach you these things in detail, but okay, still listen to me. So obviously, first of all, you will add, you will be having two different cultures, and what will you do here? You will add the stain. Which stain it is? It is crystal violet. It is crystal violet. Which stain is there? It is crystal violet, right? So the steps for this gram staining is come in and stain. C stands for crystal violet, right? Crystal violet. Is that clear? Now in I means iodine. So iodine will be added after that. Basically you can say that the stain is having crystal violet plus some potassium iodide. So iodine is here. Iodine is going to fix the crystal violet stain, right? So first of all you will add crystal violet, then what will you do? You will add iodine there. Iodine is going to fix the stain. It is like a mordant, right? It is like a mordant. It will fix the stain. What will it do, bache? What will it do? It will fix the stain. After that, you know that we will go for the washing, right? Right? So even in the lab, even if you are doing some experiments in the botany, even if you are trying to color a section there, so obviously you used to take thin sections and then you color it, right? So washing is also a steps when we talk about the staining, right? Washing is also a step when we talk about the staining. So staining can be done with alcohol, right? Alcohol or acetone. You can use anything for that washing. Right, so see, I am just telling you these steps with the help of this trick. Come in and stain. C stands for crystal violet, I stands for iodine, and stands for alcohol and acetone. So we are going to do the washing with it. Right, now look at this structure. Look at this structure. This is your bacteria. Right, look at this structure. This is your bacteria. Okay. Done. So here you have the gram positive bacteria. Just take one example like this, right? Here you have the gram positive bacteria and here you have gram negative bacteria. So now here you added crystal violet, you added iodine, you are going to do the washing. Now alcohol, it is an organic solvent. Alcohol is an organic solvent. Now this alcohol cannot create the pores here in this thick cell wall, right? Whenever we will do that step, now the step of that washing, so this cell wall in the case of the peptidoglycan layer, the peptidoglycan layer in the case of gram positive bacteria, it's very thick, right? It's very thick, right? So that washing is not going to affect here. That organic solvent is not going to affect here. It will not create the pores here. Now here, when you will do the step of washing, like whenever you will add the alcohol and acetone, now when you look at this, gram negative bacteria, so you know that inside also you are having lipid, outside also you are having lipid. So basically here lipid content is more in comparison to mucopolysaccharide. The lipid content is more. So whenever you will do the washing, so some pores will be created here. Some pores will be created here. And because we are doing the washing either with the alcohol or with the acetone, so pores will be created. So because of that, pores, cell wall is not so thick, pores are created. So stain will come out. The crystal violet stain will come out. Are you getting it? The crystal violet stain will come out. Right? Right? Crystal violet stain will come out. Okay, so no such thing will occur here in gram positive because of the thick cell wall, because of the peptidoglycan, because of that teichoic acid. So here the stain will stay like this, crystal violet. Like just take one example, outermost covering is very thick, right? Let's say you are trapped somewhere, the outermost covering, the cage in which you are trapped, it is very strong. Obviously you cannot break it and go out. If that cage is not strong, so obviously you will try, right? You will do some efforts and you can come out. Same is the case here. So in gram negative, the lipid content is more, mucopolysaccharides are less. So when there is the washing with the alcohol, the pores will be created, right? The pores will be created in these, right? The pores will be created here in these coverings, and because of that pores, crystal violet stain will come out. That color will come out, right? That color will come out. Is that clear? Is that clear? Right? So the last step here was this na, S. So S stands for saffronin. S stands for saffronin. Okay. S stands for what, bache? S stands for saffronin. So saffronin is a counter stain. It's a second stain. It's a counter stain. Now we know that we are now we know that we have the gram positive, gram negative bacteria both. In our culture, we can have both gram positive also, gram negative bacteria also. We just need to verify which one is gram positive, which one is gram negative. So obviously we are going to follow this particular procedure to differentiate gram positive and gram negative bacteria. So in first step, the crystal violet, then iodine to fix the stain, then alcohol will be used. So obviously after washing from the gram negative, the stain will come out, this crystal violet stain will come out. So we have to still see the gram negative bacteria. We still want to see the gram negative bacteria. That's why we add one counter stain, that is saffronin. So that saffronin will color the gram negative bacteria. Is that clear? That saffronin is going to color the gram negative bacteria. Is that clear? Yes. Is that clear? Sure, bache? Okay. Okay, so we will be able to differentiate, we will be able to see these two. Okay, we will be able to see these two, that this is gram positive, this is gram negative. So that is why the counter stain saffronin is used. So that is how on the basis of outer covering, that is how on the basis of the thickness of cell wall, we can even categorize bacteria into gram positive and negative. So you know that this is given in your class 11th chapter cell topic also. In chapter cell also it is given, right? This particular topic is given there also, no? So I explained it here. Okay, gram positive and negative, gram positive and negative. Done, bache? Done. So can we just revise it once again? We are talking about the bacteria. So we know that bacteria belongs to the kingdom Monera. Kingdom Monera is the only kingdom where bacteria are present. So we are discussing the structure of bacteria. We have started with the outer coverings. So we are done with the glycocalyx. So if glycocalyx is solid, if it is compact, if it is tough, it will be the capsule. If it will be jelly-like, it will be the slime layer. Next covering is cell wall. So in the case of bacteria, the cell wall is made up of peptidoglycans, also known as murines, also known as mucopolysaccharides, and some cross peptides are also there. So on the basis of these, this cell wall, on its thickness, on its, right, so we have gram positive and negative bacteria also. So we know that gram positive bacteria are the one which will give color with the gram stain. Gram negative bacteria are those which will not give color with the gram stain. Is that clear? Yes, bache, is that clear? So the next is what? So the next is the done. Next is the cell membrane. So when it comes to the cell membrane, in the case of bacteria, what do we have? Yes, what do we have when it comes to the cell membrane in the case of bacteria? Yes, bache, in the case of bacteria. So in the cell membrane will be like the eukaryotic cells, bache. It is thin, it is elastic, it is semi-permeable membrane, right? It is thin, it is elastic, it is semi-permeable membrane. It is basically a phospholipid bilayer बच्चे, right? It lacks sterols. It doesn't have cholesterol. This bacterial cell membrane, it lacks sterols, right? It lacks sterols, means cholesterol is absent there. Okay, cholesterol is absent there. Fine, cholesterol is absent there. Some hopanoids are present. Fine bache, fine bache. So cell membrane is same, it is the phospholipid bilayer. Okay, it is the phospholipid bilayer. Clear? Sure. So now come to this part. Done? So we are done with the coverings. Now let's talk about the genetic material. One by one we will discuss everything. Okay, so here you can see the complete diagram also. That will be good for you. Clear? Clear? This is the structure of the bacteria, and one by one we are discussing each and every part here. Okay bache, so now let's talk about the genetic material here. Fine, let's discuss the genetic material. Bache, in the case of bacteria, we do not have the true nucleus, right? Sometimes, you know, this type of language is given in NCERT that they do not have the true nucleus. Their nucleus is not covered with, like, see, if I have to draw it, the genetic material will be like this, right? The genetic material will be like this. It is not the true nucleus. Basically, when you talk about the genetic material, here you will be having the circular DNA, right? Here you will be having the circular DNA, right? Right. The DNA is present here. It is not linear. In the case of bacteria, it will be circular. It will be circular. Okay. It will be circular. So, DNA is present in cytoplasm and is not covered with the proper nuclear membrane, and not covered with, right, it is not covered with nuclear membrane. So when it comes to this genetic material, we call it circular DNA. We even call it naked DNA. We'll tell you the reason for it, right? Even it is known as genophore. It is known as genophore. Even it is known as prochromosome. These are the terms that we use for the genetic material. Right, these are the terms that we use for the genetic material. We even use the word circular DNA, we even use the word naked DNA, we even use the word genophore, we even use the word prochromosome. Okay, we even use the word prochromosome. Is that clear? Is that clear? So when it comes to the genetic material, as I said, there is the circular DNA. DNA is present in cytoplasm and not covered with the nuclear membrane. It is present in cytoplasm, not covered with the nuclear membrane. But it is a circular. And what is the meaning of naked DNA? Not covered with histone proteins even. Right? It is not even covered with histone proteins. This is something that you need to know, that you need to understand. So when you talk about the prokaryotes, right? When you talk about the prokaryotes, DNA is circular, not covered with histone proteins. You know about histone proteins, bache? Histone, in the case of eukaryotic cells, we discuss it, yes or no? Histone proteins. You know na, DNA. Our DNA is highly coiled, helical, very long. So it will coil over the histone proteins. Further, there will be the supercoilings, and finally it will become that compact structure. Then only it will fit in our nucleus. Then only it will fit in nucleus. Isn't it? Isn't it? Do you remember that part, Priyanka? Yes, bache, Srihara? Do you remember that? But when you talk about the prokaryotes, the DNA is not linear, it is circular, and it is not covered with the histone protein. Some other basic proteins like polyamines, some other basic proteins are present, but histone protein is not there. So because it is directly present in the cytoplasm, not covered with the nuclear membrane, we use the word naked DNA. We use the word naked DNA. We also call it as genophore, right? We also call it as genophore. Okay, 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 so one more question. Bache, something is circular. Something is circular. You can consider the example of a hollow woolen ball, right? You can even consider the example of a hollow woolen ball. So obviously it is circular. It's a circular. It's a circle. All the threads are in that circle. So basically it will be only one structure, no? It is going to be only a single structure, no? It is just a single structure, isn't it? Yes or no? Yes or no? Like if my favorite thing, let me do this. Okay. Oh, my bad. Just a minute, bache. Okay. So, see, let's say it is the DNA. It is coiled. It is circular, right? It is circular. Let's see, this is circular. It's a circle like this. That is how DNA is present. So you know that ultimately your DNA will become compact and it is going to form the chromosome. Your DNA will become compact and it will form the chromosome. Yes or no? Tell me. Yes or no? Tell me. Tell me quickly. Yes, bache, quickly. It will be having only one chromosome. It's the circular DNA. Something is in circular. It will become compact. It will become compact. It will become compact. There will be only one, right? There will be only one chromosome. Only single chromosome will be there. Same is the case here in the bacteria. Circular DNA is there. DNA, obviously, it will form just one chromosome. Mostly bacteria are having one chromosome only. So we also use the word prochromosome for it. Right? We also use the word prochromosome for it. Is that clear? We also use the word prochromosome for it. Right, bache? So one student is asking a doubt again and again, bache, gram positive will show the color, that purple color, right? But gram negative is not going to show that. It will pick up the color of saffronin. Fine, it will pick up the color of saffronin. So is that clear? The genetic material, no, it is not a proper nucleus. It is just present in the cytoplasm, not covered with a special membrane, nuclear membrane. Right, bache? Circular DNA it is, that's all. Fine, that's all. Clear? Yes, everyone, is that clear? Sure? So, even in the case of bacteria, right, we do not have but I am drawing this diagram again and again so that you don't miss anything. Okay, so please revise it even after the class. Okay, do revise it even after the class. Now, in the bacteria, we are also having some extra chromosomal DNA. We also have what? We also have the extra chromosomal DNA. What do we have? We also have the extra chromosomal DNA. So when it comes to the extra chromosomal DNA, it is plasmid. What is it, bache? It is plasmid. So plasmid is extra chromosomal circular DNA. Okay, what is this plasmid? It is extra chromosomal circular DNA, right? It can self-replicate, bache. It can self-replicate. Why do we say that it is extra chromosomal? It doesn't, first of all, it is not the main genetic material. It is other than this main genetic material, other than this chromosomal DNA plus this plasmid, it is self-replicating. It is having its own origin of replication. It can make its, right, it can make its multiple copies without the dependency on this genetic material. Okay, without the dependency on this genetic material. So extra chromosomal circular DNA which can self-replicate, right? So it gives different properties to the bacteria. Like, how come different properties to the bacteria? I'll tell you, or instead of properties, use the proper word phenotype. Different phenotypic adaptations to the bacteria. So now let's say plasmid is the circular DNA बच्चे, plasmid is the circular DNA. What is plasmid? Plasmid is the circular DNA. Obviously it will also be having the genes. It will also be having the genes, yes or no? It will also be having the genes. So now let's say if this plasmid is having antibiotic resistant genes, if this plasmid is having antibiotic resistant genes, so obviously this bacteria will also become resistant to antibiotics. This bacteria will also become resistant to antibiotics. Let's say here in this plasmid, right, drought resisting genes are there. So obviously bacteria will become drought resisting. If let's say this plasmid is having some other special property, they are having the genes which are having some other special properties, this bacteria will also have that special property. So that is why. So this is the extra chromosomal DNA known as plasmid, right? It is circular, it gives different phenotypic adaptations to the bacteria, bache, right? Because of this property of self-replication, we even use it in, right, biotechnology, right, in biotechnology. So it is, you know, that our favorite vector, it is our favorite vector, the most commonly used vector it is, right, right bache, most commonly used vector. So basically in the chapter biotechnology, you will discuss the role of this plasmid in detail. Is that clear? Yes bache, is that clear? About the genetic material, about the genetic material, about the cell coverings. Now bache, you will see some membrane like enfoldings here, right? In the case of a bacteria, you will not see such structures in the case of eukaryotes. In the case of bacteria, when you talk about the cell membrane, some forms some inner enfolding, some finger like projections will be formed here, right? We used to call it as mesosomes. What we used to call it? We used to call it as mesosomes. So now we'll be talking about the mesosomes. Okay, so bache, one by one we will cover everything, don't worry. So what will be discussed? We will discuss mesosomes. What will we discuss? We will discuss mesosomes. So mesosomes are basically the finger like enfoldings of the cell membrane, right bache? This question can be asked in paper. These are finger like enfoldings of cell membrane. What is it? It is the finger like enfolding of cell membrane that increases the surface area as well for the exchange, obviously, that even increases the surface area. There are different roles of mesosome. We have to discuss each and every role. It is important. So when you talk about the mesosomes, what are they? They are the finger like enfoldings. Right bache, these are finger like enfoldings present in the cytoplasm. These are the extensions of your plasma membrane. Okay, okay, finger like enfoldings they are. They are not just finger like, right? They are not just finger like, no doubt here. Like here you guys can say these are finger like enfoldings. Obviously they are lying in the cytoplasm. They are the extension of plasma membrane, that we can see. And here, you know what, in mesosomes you will also be having the respiratory enzymes, right? You will be having the respiratory enzymes, right? But it is not in that form only. Mesosomes are not just like that only. I will discuss the role as well. Mesosomes can also be there. Vesicle form. Vesicle, you know na, some rounded structure, right? You know na, vesicle, some rounded structure. So it can be there in the form of vesicles. It can be there in the form of tubules, some tube like structures. It can be form of lamellae, like some thin, something like this. Some tubes. It can be branched also like this. So it can have different structures as well, different forms, bache, right? It can have different forms. So when it comes to the function of the mesosome, right? When it comes to the function of the mesosome, bache, right? So it helps in cell wall formation. This is important. And you will not see such structures in the case of eukaryotic cell. It is unique to this. So when you talk about the mesosomes, it helps in cell wall formation. It helps in the DNA replication. Okay. it helps in the DNA replication as well, it helps in the cell formation as well, it helps in the respiration as well. Now see bache, you know that prokaryotic cell, bacteria, we are discussing the kingdom Monera. Bacteria is the sole member of kingdom Monera. So bacteria is the sole member means bacteria is the only member of kingdom Monera. So when it comes to the bacteria, the prokaryotic cell, you know that it is very primitive cell. It is not having the proper nucleus, it is not having the organelles, right? It is just a very simple, very primitive structure. Now you know that when you talk about the eukaryotic cell, we even discuss the mitochondria there, isn't it? We discuss the mitochondria there, the powerhouse of cell, which is taking role in the ATP, which is playing role in the ATP formation, which is having the electron transport chain. But we do not have any such organelle here in the bacteria. We do not have any such organelle here in the bacteria. So if we do not have any such organelle here, so how do these bacteria will respire? How? So mesosome is going to play a role there. So mesosome is having the respiratory enzyme, so it is playing role just like mitochondria there, right? It is playing role just like mitochondria there, right? So cell wall formation, DNA replication, even in the respiration, and moreover in the secretion process, in some secretions also, in the secretion process your mesosome play the role, right? It increases the surface area of the plasma membrane. I told you already, it increases surface area of plasma membrane. So obviously even it is also increasing the enzymatic content, right? The enzymes which are present there, obviously it will also be increased. Done. So this is the role of mesosome. So in this kingdom Monera, when we talk about the reproduction of bacteria, there also you will see the DNA will replicate, mesosome will start dividing, and then mesosome will divide the bacteria into two. So it also helps in the cell wall formation, in the DNA replication it is also playing the role. So the function of mesosome is very important. It is itself a very important question, bache. It is itself a very important question. Is that clear? Yes everyone, is that clear? Yes bachcho, is that clear? Tell me quickly. Done? Sure? Sure. So this mesosome, right, this is extra point. You want to write it, you can. You don't want to write it, it's your wish. So mesosome is well developed, right? This mesosome is well developed in gram positive bacteria. It is highly developed, you can say, in gram positive bacteria in comparison to gram negative, right? So in gram positive bacteria it is highly developed in comparison to the gram negative bacteria. What are we discussing? We are discussing the mesosome. And how can I forget to tell you this genetic material we are discussing now? So you know that here for the prokaryotes we use the term nucleoid, right? Nucleoid. So nucleoid, the naked DNA, the circular DNA which is not covered with the histone proteins, directly there in the cytoplasm, not covered with the nuclear membrane, right? So mesosome, the genetic material, the plasmid. Is that clear, bache? Yes, is that clear? Is that clear? Yes or no? Tell me, bache. Sure? Okay. So next thing that we need to discuss here in the case of bacteria is, bache, see, no doubt, as I said, the structure of bacteria is very simple. The structure of bacteria is very simple. It is not complex at all. It doesn't have the organelles, well developed membrane bound organelles. It is having this membranous extension, right? We used to call it as mesosome. We have already discussed it. We have already covered the extracircular DNA, the plasmid. We have already discussed about the nucleoid. Now in the case of this bacteria, there is only one organelle, right? There is only one organelle. What do we have here? We just have only one organelle. And what is it? It is the ribosome. It is the ribosome, right? In the case of bacteria, in the case of prokaryotes, you just have one organelle, right? You just have one organelle, and it's a non-membrane bound organelle. It's a non-membrane bound organelle, right? It is not covered with any membrane. Ribosome, always remember this thing, bache. It is not covered with any membrane, and it is even considered as universal organelle because it is present in prokaryotic cell as well, it is present in eukaryotic cell as well. Okay, it is present in prokaryotic cell as well, it is present in eukaryotic cell as well. So it is a non-membrane bound organelle, it is the universal organelle. And bache, in the case of prokaryotes, you have 70S ribosome. What do you have there? You have 70S ribosome. S here is the sedimentation coefficient. What is S here, bache? S here is the sedimentation coefficient, right, bache? It is basically the Swedberg unit. What is it? It is the Swedberg unit. Yes, bacho, what is it? It is the Swedberg unit. Everyone with full josh, with full energy. Right, it is the Swedberg unit. So basically, when we study the structure of a cell, we go for the technique cell fractionation. Right, we divide the component of cell in fractions. Obviously, we will use the ultra centrifuge. So basically, as per the weight, the things will settle down. The thing which is very heavy, it will settle down at base. The things which are having, which are having lighter weight, so obviously they will come at the top. So this S is telling us about that sedimentation coefficient. So Swedberg unit, this is what we are discussing. So it is basically the Swedberg unit. So in the case of prokaryotes, you have the bacteria, you have the ribosome, the 70S. So this 70S ribosome is made up of two things. One is the larger subunit, bache, and another is the smaller subunit. Right, one is the larger subunit, another is the smaller subunit. So larger subunit is 50S. S is again the sedimentation coefficient. Smaller subunit is the 30S. Larger subunit is the 50S. Smaller subunit is the 30S. Are you getting my point? Are you getting my point? Larger, smaller, larger, smaller. So bache, there is no need to add it that 50 plus 30 is 80. No, no, no. Here we are just talking about large and small subunit, right? Right? There is no need to add it that 50 plus 30. No, it is as per the sedimentation, na? It is as per the sedimentation, as per the weight it is. Fine, it is as per the weight. So larger, smaller, larger, smaller, larger, smaller. So 50S, 30S, 50S, 30S. Clear? 50S, 30S. Done? Done? So in the case of these ribosomes, as I said, in the case of prokaryotes, they are 70S. The larger and smaller is there, but bache, they will not always stay connected to each other. No. So they will join only at the time of protein synthesis. And how will it join? With the help of magnesium ion. Right? So magnesium ion will help in the joining of the large subunit to the smaller subunit. I always give this example, right? Like in our home, we don't talk generally with our siblings, no? We used to have that cold war with our siblings. So we generally don't talk to them, but whenever any relative come, our mother used to say that don't you dare to fight in front of relatives. You have to stay together. At least you have to show that you are together, right? So same is the case here. So relative is what? The protein synthesis. So whenever there is the requirement of protein synthesis, at that time what will happen? The large and the small subunit will join together. Okay, the large and the small subunit will join together. And how will it join? It will join with the help of mummy. Mummy here is M, that is magnesium. Right? Mummy here is M, that is magnesium. Clear, bache? Clear, bache? So bache, today's lecture is very informative. Right? Whatever point I am highlighting now, that this is important, they all are PYQs. They all are previous year questions. Fine? Fine. So larger and smaller subunit will join only at the time of protein synthesis. Now in the case of prokaryotes, what is the scenario, bache? In the case of prokaryotes, because there is no nucleus, right? In the case of prokaryotes, because there is no nucleus, right? So here in this case, the transcription and translation is occurring simultaneously. In their case, the transcription and translation is taking place simultaneously. Means the formation of mRNA and protein is taking place simultaneously, means altogether. So one mRNA, when so many ribosomes are attached, and simultaneously when protein synthesis is going on, right? So basically in prokaryotes, transcription, translation is taking place simultaneously, and here when one mRNA is attached to the many ribosomes, right, one mRNA when it is attached to the many ribosome, we used to call this structure as polyribosomes or polysomes. We also used to call it as polyribosome or polysomes. Are you getting it? Are you getting it? We even used to call it as polyribosomes and polysomes. This is what you will see in the case of ribosomes. So ribosomes are what? They are the protein factory. They are going to form the proteins, right? They are basically the protein factory. They will form the proteins. So it's a non-membrane bound organelle which is present in a prokaryotic cell. So it is the only organelle which is present in a prokaryotic cell, right? It is the only organelle which is present in the prokaryotic cell. So you guys can see in the prokaryotic cell there is no, right, there is no organelle. There is no membrane bound organelle. Only outer covering is there. Only these three layers are forming the outer covering, yes or no? Only these three layers are forming. Kartika bache, only these three layers are forming outer covering, yes or no? Yes or no? So you know that that is why in the case of prokaryotes we used to say that they have one envelope system. What do they have? They have one envelope system, means they just have the outer coverings only. Okay, they just have the outer covering only. Fine, fine bache, fine. Okay, so in the case of prokaryotes, you even have some chromatophores. Bache, we need it. In the kingdom Monera, we need it. Right? So listen to me carefully. So chromatophores are also there in a prokaryotic cell. Chroma means color. So basically what is happening in bacterial cell? Okay, in bacterial cell, some membrane extensions are forming some sac-like structures, right? They are forming some sac-like structures in the case of prokaryotes, right? Some sac-like structure in photosynthetic prokaryotes, right? In photosynthetic prokaryotes. Photosynthetic prokaryotes means your blue-green algae, your cyanobacteria. Are you getting it? So basically in the case of prokaryotes, you are having some membrane bound structures, some other sac-like structures, right? You will see it in the case of photosynthetic prokaryotes, that is your cyanobacteria, that is your blue-green algae, right? So in their case, these these sac-like structures, they are having some pigments which will help in the photosynthesis, okay, right, which will help in the photosynthesis. So chromatophores are also there. Fine bache, chromatophores are also there. Is that clear? Is that clear? Sure. So even inclusion bodies are also present in the case of prokaryotes. This is also a PYQ. It is something unique which is present in the prokaryotes. Like you will not see mesosomes, you will not see inclusion bodies in the case of eukaryotes. You will see such structures only in the case of prokaryotes. So when you talk about the inclusion bodies, bache, right, these are the storage granules. These are the storage granules present in cytoplasm, right? These are the storage granules present in cytoplasm and they are without membrane. they are without membrane. These are basically the storage granules. Like in the cytoplasm, some storage, stored food is available. So like you will be having the glycogen granules like this, right? Some storage granules are present in the cytoplasm, which are basically the inclusion bodies, right? They are not covered with any of the membrane. Fine, bache, they are not covered with any of the membrane. Fine, okay, so glycogen granules, they are the examples, right? Glycogen granules, they are the examples. Clear, bache? And along with that, in some bacteria, gas vacuoles are also present. Again, they are not covered with any membrane, bache, right? They will provide the buoyancy to the bacteria. Okay, will help in float in that bacteria, will help in floating, right? So yes, along with the glycogen granule, you have some synophysian granule, you have some phosphate granules. Fine, so these are the inclusion bodies. Done, bache, these are the inclusion bodies. So this is about the structure of bacteria. Only two things are left. Let's discuss it and then we will start the kingdom Monera in detail. We have almost covered everything there. So now in the case of bacteria also, there is a flagella. If the bacteria is motile, if it is movable, it will be having the flagella. Yes, after it I will give you the break, I will take your doubts, okay? After 10 minutes now, I'll give you the break, I will take your doubts. So obviously if it is a mobile bacteria, if it is a movable bacteria, if it can move, then it will have flagella then. So here the structure of flagella in the case of bacteria is very simple, bache. It is not just like eukaryotic cell. It's a very common question, right? It is not just like eukaryotic cell. It's a very common question. Here the flagella is made up of, right? It is made up of flagellin protein. It is made up of what? It is made up of flagellin protein. Are you getting it? It is made up of flagellin protein. Now if you compare it with the eukaryotic flagella, wait, I will write it here. Right, do include it in your notes, bache. This is something very important. Right, when you talk about the eukaryotic flagella, it is having tubulin protein. It is having microtubules. Okay, it is having microtubules, the tubulin protein. But when you talk about the flagella in the case of bacteria, in the case of prokaryotes, what will you have? You will be having the flagellin protein. Right, you will be having the flagellin protein, and the bacteria here is very, very, very simple. Fine, bacteria here is very simple. Okay, so in the plasma membrane, there will be the basal body, there will be one hook, and there will be this thin filament. Okay, only three components will be there in the bacteria. In the case of prokaryotic bacteria, there will be only three components. The one is your basal body, the hook and the filament. The hook and the filament, that's all. So basal body, the base from where the flagella will arise, the hook-like structure will be there, and then there will be the thread-like. There will be the thread-like, right? The longest part, the filament will be there. Is that clear, bache? Is that clear? Yes or no? Is that clear? So this comparison is important, bache. Don't be like that, ma'am, you are mixing two different chapters. Why are you explaining the structure of prokaryotes here? Because see, we will complete it here in biological classification. So in the chapter cell, we will directly start from the eukaryotic cell. There will be no need to discuss the prokaryotic cell there. Okay? Okay, and even you guys will be able to relate it with the kingdom Monera. So kingdom Monera, it will be easy for you to remember the kingdom Monera there. Right, so after finishing this, we just need 30-45 minutes to complete the kingdom Monera. Okay, we just need 30-45 minutes to complete the kingdom Monera. I know it is very difficult for you people to discuss biological classification for such a long time, but actually it is a lengthy chapter, bache. If anyone can claim that we can finish it in 2 hours, 3 hours, means they are just like 3 hours are still fine, but they are just giving you the overview, right? Right, so if you are doing it for the first time, because now we have the time, we don't have 3 months or 2 months for the NEET. We have sufficient time, and in that sufficient time we should clear our concepts, We should focus, right? No doubt we have to complete the syllabus as well, but right, we have to understand that syllabus. This should be your agenda. Okay, this should be your agenda. Done. So basal body, hook and filament, these are the components when you talk about the prokaryotes. Clear bache? So even in prokaryotes, some vessel like, okay, even in prokaryotes, they contain some non-motile structures also. Some non-motile structures. Okay, and that is what? Fimbriae and pili. Right? Fimbriae and pili. Pili is made up of pilin protein. They will help in the attachment to the substratum. Like if I talk about the fimbriae, it helps in the attachment to substratum, like to the rocks, right? If say that bacteria is in a aquatic medium, so it will help in getting attached to the rocks. And moreover, bache, these are small, they are non-motile, they are playing no role in the movement, but they are playing role in the attachment, right? This is an important question. Pili and fimbriae, they are what? Pili and fimbriae, they are what? They are non-motile structure. They will not play any role in the movement, they will help in the attachment. Like in the case of fimbriae, it also help to get attached to host cell, right? It also help to get attached to host cell, to the host cell. Like I always give you the example of Neisseria gonorrhea. Remember this bacteria, bache? Neisseria gonorrhea. Neisseria gonorrhea. It used to cause sexually transmitted disease. That is gonorrhea. This is a bacteria, right? It is a bacteria which used to cause a sexually transmitted infection. That is gonorrhea. So this bacteria, with the help of this fimbriae, right? This bacteria, with the help of this fimbriae, it helps, it get attached to the host cell. It get attached to the host cell. Okay? It get attached to the host cell. Now, when it is the pili, this one, now these are some tube-like structure. Like let's take the example of E. coli. Let's take the example of Escherichia coli, the world famous bacteria, isn't it? So they are also having some tube-like structure. So this tube-like structure will help this bacteria to get attached to the another bacteria, right? This tube-like structure will help this bacteria to get attached to this another bacteria. So here in this bacteria, there is one plasmid, the F plasmid. Right, the fertility factor is there, the F plasmid is there, and here in this bacteria, F plasmid is absent. So this is F positive plasmid. F means fertility. F means fertility. So here in this bacteria, we have some fertility factor. F plasmid is present here. This bacteria is F negative because here this fertility plasmid is absent. Here this fertility plasmid is what? It is absent. So what is going to happen here, bache? With the help of this conjugation tube, right? With the help of this conjugation tube, this F plasmid will get transferred here, right? And this bacteria will also become F positive. So this pilus, pilin, right? Pili is plural, pilus. Pilus is singular. So basically it's a kind of conjugation tube. It helps in the reproduction. It's not a proper sexual reproduction, but in bacteria we will see such cases. We will see conjugation, so it helps there, right? Sex pilus is there, you are right. So these are basically the non-motile structure. What are they? These are basically the non-motile structure. So this is about the structure of bacteria, bache. I hope the structure of bacteria is crystal clear to you, right? I hope the structure of bacteria is crystal clear to you. Tell me, is it? If there is any doubt, do let me know. Non-motile means it is not playing any role in the movement, right? It is playing no role in the movement. Clear, bache? Yes, is it clear? Sure? Okay. Sure, right? So this is about the structure of bacteria. Now we will discuss the shape of bacteria. Can you tell me anything about the shape of bacteria? And why are we discussing it? Basically, we are discussing the kingdom Monera. Kingdom Monera is having what? Kingdom Monera is having the bacteria. Bacteria are the sole member of kingdom Monera. Bacteria are the sole member of kingdom Monera. Okay, so we have completed the structure of that bacteria. Now we will talk about its shape. What will we discuss? We are going to talk about its shape. So the very first thing is coccus, cocci, right? So coccus is singular. Coccus is singular, bache, right? Shape. Then you know that Vibrio, the comma shaped bacteria, right? Then you have the Bacillus, the rod shaped bacteria, the cylindrical, right? Here it is the spherical, and then one is the spirillum, spiral shaped bacteria. So bacteria, they are having different shapes as well. Clear bache? They are having different shapes as well. Is that clear? Yes bacho, is that clear? Right? Is that clear? So different shape is there. And now if I talk about the flagella, so it is atrichous. Atrichous. Bacteria is atrichous if flagella is absent. If flagella is absent. Okay. And it is monotrichous if flagella is present. If flagella is present, but it is only one flagella is present. Clear bache? Monotrichous. So you have other words also, cephalotrichous, amphitrichous, right? So let's say if it is amphitrichous. So trichous word is basically related here to the flagella, the attachment to that flagella. So amphitrichous means, let's say this is the bacteria, both sides are having the flagella, right? Both sides are having the flagella. So this will be your amphitrichous bacteria, right? Right? Both the sides will have the flagella there. Okay, bache? Okay. So this is about the shape of bacteria. This is about the shape of bacteria. And here it is about the insertion of flagella. Atrichous without flagella, monotrichous only one flagella is there, amphitrichous when both ends contain flagella. When both ends contain flagella, then it will be amphitrichous. So any doubt? Yes bacho, any doubt? Yes? Do you have any doubt? Sure? Done बच्चे? Good बच्चे Samia. Okay. So do you want break? Do you want break? But that break will be for just 10 minutes, or you can say that 11 minutes. Okay, so there will be the break time. Now there will be the break time. Okay, so break time is from 8:50 to 9 PM. Clear? 8:50 to 9 PM. So after that we will again continue Kingdom Monera. Now we have to discuss the reproduction in Kingdom Monera and the nutrition. Once it's done, we will individually talk about the mycoplasma, the photosynthetic bacteria, all that, you know, Nostoc, heterocyst, like this. So this is what we need to cover. Okay, this is what we need to cover. So just Monera we will complete today, right, bache? just Monera we will complete today, and for that Protista and fungi, because it is not very lengthy. Today we have completed the introduction part or the Monera. We will discuss it in the next marathon. Okay, we will discuss it in 2 parts, and on Friday, today it's Thursday, yes, means tomorrow, there will be the question practice session from the living word as well and from the biological classification as well, but majority of the questions will be from the kingdom Monera only. Is that clear? Majority of questions will be from the kingdom Monera only. So as of now, you guys will get a break for the 10 minutes only, right? For the 10 minutes only, there will be a break, and then we will continue. So till then, take care. Bye-bye. Hi everyone. Can you hear me now? Hello, hello, hello. Yes, can you hear me now? Okay, so let's continue, bache. So we have covered the structure of the bacteria, the shape of the bacteria, and about the flagella. So now we will start the nutrition and the reproduction in the case of bacteria. So basically we are talking about the kingdom Monera. So in the kingdom Monera, the sole member, the only member is the bacteria. So we have to discuss everything related to the bacteria. Okay. Okay, so now let's talk about the nutrition here, bache. Let's talk about the nutrition. So you know that when we talk about the R.H. Whitaker system of classification, he even considered the mode of nutrition to classify the organism, right? He even considered the mode of nutrition to classify the organism, right? Right. So when it comes to the nutrition, we have autotrophic and the heterotrophic. We have autotrophic nutrition and the heterotrophic nutrition. So bache, I have added this okay, so basically I have added this picture from a book, right? So it will explain everything, right? It will explain everything you can see here. So when it comes to the nutrition in the case of bacteria, we have the autotrophic bacteria as well. We have the heterotrophic bacteria, right? We have the autotrophic bacteria as well. We have the heterotrophic bacteria. Let me write down. Guys, please take the notes. So one is what? Autotrophic and another is heterotrophic. So you know the meaning of autotrophic, isn't it? You all know the meaning of autotrophic nutrition, isn't it? Auto means self. This bacteria is capable of making its own food, right? Auto means self. This bacteria is capable of making its own food. So here you have two examples. One is your photosynthetic bacteria, that is your blue-green algae, that is your cyanobacteria, right? And another is your chemosynthetic bacteria, right? It is going to use the chemicals. It is going to use the inorganic compounds for synthesizing its food, right? Another will be chemosynthetic bacteria. Now when you talk about the heterotrophic, so obviously you will be having the saprophytic. Heterotrophic means they cannot make their own food. They are dependent on other organisms, isn't it? So when it is the heterotrophic bacteria, saprophytic it can be parasitic, and bache, one more category is there. It is saprophytic, it is parasitic, one more category is here. Can you tell me? Autotrophic means those who are going to make their own food. So we have two examples. One is of photosynthetic bacteria, that is your blue-green algae, and another is of chemosynthetic bacteria, that is here the chemical energy will be used. Basically inorganic compounds will be used. So here what do we have? One is saprophytic, another is parasitic, and the third category will be symbiotic. Exactly, symbiotic. So bache, understand the literal meaning of these terms. So when it is saprophytic, means it depends upon the dead and decaying organic matter. These are the bacteria which will feed on dead and decaying organic matter, right? Dead and decaying organic matter. This is the meaning of the saprophytic bacteria. Now when it is parasitic, parasite, it needs another living organism. It needs a host body from where it can take its nutrition. Parasitic it will be. Now it is symbiotic. Again, it requires the host. It will live with that host. Both are dependent on each other, isn't it? Both are dependent on each other, isn't it? Isn't it, bache? Isn't it? Symbiotic. Okay. Now, photosynthetic, chemosynthetic. Yes, bache, we do have the bacterias. In which photosynthesis is possible. We will discuss about it. We will take the example of nostoc, right? Anabina. We are going to discuss that examples also. But here when it is the photosynthetic bacteria, right? Here when it is the photosynthetic bacteria, it is not going to use the H2O as electron donor. This is what you need to remember, right? This is what you need to remember. In the case of photosynthetic bacteria, it is not mandatory that all the bacteria will release oxygen. Do you know about it? Do you know about it? Right? Before this, I'll explain you something else. Do you know that some bacteria are aerobic bacteria and some bacteria they are anaerobic? Are you aware of it, bache? Do you know about these terms? That some bacteria they are aerobic and some bacteria they are anaerobic. They are anaerobic. So when it is aerobic bacteria, it lives in presence of oxygen. It needs oxygen basically. So it lives in presence of oxygen. And when it is anaerobic bacteria, it doesn't require oxygen. Right? It doesn't require oxygen, isn't it? It will be anaerobic bacteria, right? So same way we have few terms, right? Like we have facultative. See, on the basis of it, these are two general words, aerobic, anaerobic. Aerobic, which can survive in oxygen, which needs oxygen basically. Anaerobic, which does not require oxygen, right? Which does not require oxygen. So now on the basis of it, we have two terms. One is obligate, another is facultative. One is obligate, another is facultative. Do you know the meaning of these two terms, bache? One is obligate, another is facultative. Firstly, I'll explain it and then we will come to the nutrition part. One is obligate, another is facultative. Obligate, very good, Smiha. O for obsession. O for obsession. This is how I remember it. Obsession. You are obsessed for something, means you need that particular thing, isn't it? When we say O for obsession, means you need that particular thing. You want that particular thing. So when it is the obligate bacteria, when we use the word obligate, means it needs that particular thing. Like for an example, if I'm saying obligate aerobes, right? Let's say if I'm saying obligate aerobes, means the bacteria which cannot survive without oxygen, it needs oxygen only. Right? This bacteria which cannot survive without oxygen, it needs oxygen. It is obligate aerobe. It will not survive without oxygen. Even we are obligate aerobes. We cannot survive without oxygen, isn't it? Yes or no, bache? Bache, see, your energy should be very high. So please focus here, right? So they need something very strictly. Now when it is facultative, it's simple. Facultative, like if they'll get oxygen, they are okay. If they are not getting oxygen, still they are okay. Right? Still they are okay. Okay, so obligate means strictly we need that particular thing. Just like in the case of host and parasite relationship, we used to say that some parasites, they are so specific, they need that particular host only. Otherwise, they will die. So obligate, right? Obligatory relationship is there. Obligatory relationship is there, right? So when it is facultative, it is not totally dependent on that particular thing. If they get it, good. If they don't, again it's good. So one word is obligate aerobes. Let's say now another word is facultative aerobes. So now can you tell me the meaning of facultative aerobes? Facultative aerobes means bacteria live without oxygen. Bacteria live without oxygen, but can survive in oxygen too. Like, so it is facultative aerobe, means it is not totally dependent on oxygen. It usually lives without oxygen, but it can survive in oxygen too. It can survive in condition where oxygen is present. Got it? So same is for the obligate anaerobes. They don't need oxygen at all. If there will be oxygen, they will not survive. Same way, facultative anaerobes. They live in oxygen, but if they are not getting oxygen, still they are okay. Still they are okay. Facultative anaerobe, right? So now are you able to understand? Obligate aerobe means for sure it needs oxygen. It cannot survive without oxygen. Facultative aerobes means normally it was living without oxygen, but if it will get oxygen, still it can survive, right? Still it can survive. Is that clear? Yes, bacho, is that clear? quick. Tell me quickly. Then I'll give you the homework. Right now we are talking about the respiration and then we will talk about the nutrition. Because somewhere it is related, that's why I'm telling you this. Okay? Sure? So strict means obligate, facultative means it's okay with everything. So this is about the respiration. Now, you know that respiration, right? Respiration, you know that it is a very important process. Ultimately, we are getting the oxygen, we are using that oxygen to break the food, to break the glucose, and the energy will be released, right? So it is a something very important. Now, obviously, we can relate the nutrition with it. So, bache, when the word is autotrophic, auto means self. So obviously this bacteria will make its own food. Like the best example is photoautotrophs. Photoautotrophs means in the presence of light, right? In the presence of light, it will make its own food. In the presence of light, it is going to make its own food. Is that clear? Same way, we have chemoautotrophs. In the presence of chemicals, it will make its own food. Depend upon, nitrates, nitrites, ammonia, right? They don't need light for making their food. They are dependent on nitrites, and they are dependent on nitrates, ammonia, like this. Now, when it is the photoautotrophs, there is no need to remember everything written here, but I think you all have studied this particular example in your NCERT, green sulfur bacteria. See, bache, when the word is photosynthetic, see, some bacteria, in the case of bacteria, it is not mandatory that they will produce oxygen. No, bacteria can be non-oxygenic. It can perform non-oxygenic photosynthesis. It can perform oxygenic photosynthesis, right? It can form non-oxygenic photosynthesis. It can form oxygenic photosynthesis, right? Non-oxygenic or oxygenic. Are you getting my point? So non-oxygenic photosynthesis means it will not release oxygen. It is not going to release the oxygen. Oxygenic photosynthesis means it will release the oxygen. Now, it depends upon two things. But whatever I am teaching you here, it is somewhere related with the other chapters in your diversity, in your NCERT, in your class 11 syllabus. So, please listen to me very carefully. So, one word is non-oxygenic photosynthesis, another word is oxygenic photosynthesis. So, what am I saying? That when you talk about the bacteria, it is not mandatory that every time while doing the photosynthesis, it will release the oxygen. No. It can be without oxygen also. Why? Because in such photoautotrophs, they are not using hydrogen as the electron donor. They are not using hydrogen, right? Instead of that, let's say they are using H2S. So, CO2 plus H2S. So, obviously, there will be the release of sulfur. It is given in that photosynthesis part, yes or no? There will be the release of sulfur, right? I am not balancing the carbon number and everything here, right? So simply you can even write the word sugar instead of glucose, okay? So sugar and the sulfur will be released. So it will be the non-oxygenic photosynthesis where oxygen will not be released because we are not using, the bacteria is not using the water here. Bacteria is using another donor here, right? Another donor here, and that is the H2S. Bacteria is using another donor here, and that is what? That is the H2S. Is it clear? Yes, bache, is it clear? Yes or no? Yes or no? Right? Even in the chapter photosynthesis, we will discuss it. So there is no splitting of water here. There is the splitting of H2S. This is the electron donor. It will basically reduce your carbon dioxide. So in oxygenic photosynthesis of bacteria, H2O will be used only then, because there are some aerobic bacteria, they will use H2O, only then they will release the oxygen. Okay? Okay, these terms are important one way or another way. They are related to your syllabus. That's why I'm telling all these things. That's why I'm explaining this. So, photolithotroph bacteria, basically green sulfur bacteria, your purple non-sulfur or green non-sulfur, they all are the example of photoautotrophic bacteria. Okay, and when it is the chemoautotrophs, right, they are not dependent on light for cooking their food. right, for making their food. They are not dependent on light, they are dependent on chemicals. Like see, nitrifying bacteria, right, iron bacteria, hydrogen bacteria, sulfur bacteria will be there, right, sulfur bacteria will be there. So they are using, this bacteria is using H2S, right, this bacteria is using ammonia here. So these examples are important, bache, these examples are important. Example of nitrifying bacteria like Nitrosomonas, Nitrococcus, right, we discuss it in plant physiology. Then Nitrobacter, Nitrocystis, okay, right, ammonia to nitrate, nitrate to nitrite, done. So is that clear? Is it clear? So these are the different ways of nutrition in the case of bacteria. Is that clear, bache? These are the different ways of nutrition in the bacteria. Now next is the reproduction. Next is what? Next is the reproduction. So reproduction in Monera means reproduction in bacteria. So bache, see, majorly it is asexual reproduction. Majorly it is what? It is asexual reproduction. Are you getting it? Majorly it is asexual reproduction. The sexual reproduction is also there, but it is not proper sexual reproduction. The genetic recombination will take place, no doubt. The genetic recombination will take place here, but it is not the proper sexual reproduction. There is no gamete formation or something. No, the conjugation, the transformation, such things will be here. Okay, so when it is asexual, the very common way is binary fission. Very common way is binary fission, and then there is the spore formation also, which is known as sporulation. Which is known as sporulation. Okay, which is sporulation. So see, as I said, the common method here is fission. Then the sporulation, spore formation is also there. When it is the genetic recombination, the sexual reproduction, so you will be having the conjugation, transformation, transduction. So these are the different ways that we discuss in the case of sexual reproduction in the case of bacteria. Okay, so asexual is binary fission and sporulation. Asexual is binary fission and sporulation. And bache, in the case of bacteria, you know, the division is very simple. It is not like mitosis. It is not like mitosis. You know what is going to happen here? What is going to happen here? So very first thing is the DNA replication. The DNA will replicate, then your mesosome will start dividing. Okay, the very first step here is the DNA replication. So when we talk about the sexual reproduction in the case of bacteria, so the very first thing that will occur here is what? Right, the binary, the DNA replication, and then what will you see? That mesosome will also start growing. Okay, the mesosome will also start growing. Wait, I'll explain it. Okay, okay. So basically, your bacteria, it will start its elongation. So mesosome formation will be there. Okay, mesosome formation will be there. Right. So the very first step here is the DNA replication. Then what is going to happen, bache? What is going to happen? The mesosome formation, or you can say that the mesosome division will be there. Right. The mesosome division will be there, and the cell wall will also form. Mesosome is helping in the cell wall formation, na? And finally the bacteria will divide. It is very simple, not like mitosis, the prophase, metaphase, anaphase, telophase will be there. No, 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 no. No, nothing like this will occur. Nothing like this will occur. So simply bacteria will elongate, then your mesosome will divide, even your cell membrane, not the cell wall. Firstly cell membrane and then the cell wall will form, and then it will divide into two daughter cells. That's all. It is going to divide into two daughter cells. That's all. Simple. Simple it is. Okay, finally the two daughter cells will divide, right? Finally, the two daughter cells will divide. So it is going to be very easy, right? It is going to be very easy. Here also you guys can see. So basically the DNA will replicate, bache. The cytoplasmic membrane, it will elongate, right, bache. The cytoplasm divides and the cross wall will form. So cytoplasm will divide and then the cross wall will form, and there will be the two daughter cells, right? There will be the two daughter cells. What will be there? There will be the two daughter cells. Fine, two daughter cells. Clear, bache? Clear? And you know that sporulation. Do you know about the sporulation? What is the meaning of sporulation? Yes, bache. Sporulation means spore formation. So you know that in adverse conditions, sometimes in the case of bacteria, there is the formation of spore. Some endospores will form, right? Some endospores will form. Fine, bache. So this is the sporulation. What is it? It is the sporulation. So when conditions are not favorable within the bacteria, the endo means inside, the spore will form, right, bache? So when the conditions will be favorable, the bacteria, the breakdown of cell wall will be there, endospore will again grow. Fine? Done, bache? Done? Okay, sure. And then here the conjugation. I have already explained you the conjugation. Remember I told you in the case of E. coli, there is the formation of the sex pilus, right? There is the formation of the sex pilus. Basically in bacteria you will be having the circular DNA, right? And moreover here we will see one plasmid, right? So this plasmid is F plasmid. So if this bacteria is having F plasmid, we used to say it F plus, right? This F means fertility. And this bacteria can pass this plasmid. It can duplicate this plasmid and it can pass it to another bacteria which is not having this particular plasmid, okay? So this is conjugation. So obviously genetic recombination is there. A part of it will pass here, it will grow here. Okay, a part of it will pass here and it will grow here. So this is basically conjugation, and here this sex pilus will form this conjugation tube. Are you getting it? This sex pilus will form this conjugation tube here. So this is conjugation. So one F plus plasmid, when F plus bacteria can convert another F negative bacteria into F plus bacteria. Are you getting this part? Yes, bache, this is the conjugation. Right, this is the conjugation. So this tube is the conjugation tube which will bring these two bacteria close to each other. Clear, bache? Clear, bache? So after it, this bacteria will also become, both the bacterias will be like this, having fertility factors. So of course there is the genetic recombination, right? So it is not a proper sexual reproduction, but again you can see it is a sexual reproduction. So this is the conjugation. Now next is bache transformation. Can you tell me what is transformation? Can you tell me what is transformation? We will even discuss it in class 12th syllabus, right? We are even going to discuss it in class 12th syllabus. In class 12th syllabus we talk about the Griffith's experiment, na? We talk about the Griffith's experiment. Do you remember that? Griffith's experiment. Like let's say, what is transformation? Let's say this is a bacteria. In simple words I am explaining it, bache. In its environment, in its surrounding, it is having some other genetic material. Okay. If that genetic material, it is entering here in this bacterial cell, it is getting incorporated here in the bacteria's genome. Right, so obviously whenever this bacteria will divide, right, it will have this outside genetic material also. Yes or no? Transformation, I am explaining transformation. It is also a mean of sexual reproduction in Monera, in kingdom Monera, in bacteria. So we are talking about the Griffith's experiment here, right? So in Griffith's experiment also we discussed now that there is a bacteria R strain, and the S strain is there. So when you will heat kill the S strain, and in the same mixture when you have the rough strain, so rough strain can pick up that virulent DNA from the surrounding. So bache, we will discuss it in class 12 syllabus. So as of now, what is transformation? When a bacteria, it picks up the genetic material from its surrounding and it gets incorporated here in its genome. So whenever this bacteria will divide, obviously that bacteria will also contain that surrounding wala genetic material. So obviously it will add, it is going to add new genes here. It is also a kind of genetic combination, isn't it? Yes or no, bache? Tell me, are you able to understand? Yes or no? If no, then please tell me, I'll explain it. So transformation is when bacteria is picking up the genetic material from surrounding, right? When bacteria is picking up the genetic material from surrounding, and that genetic material is getting attached to its genetic material. So when the bacteria will divide, obviously that surrounding, the genetic material which was there in the surrounding, it will also become the part of bacteria's DNA. It will also become the part of integral part of that bacteria's genome. Is that clear? That is the transformation. And next is transduction. Next is what? Next is transduction. What do we have? We have transduction. So bache, transduction is when we use another agent to pass the DNA, to pass the genetic material to a bacteria. Like let's say this is the bacteria that we have. This is the bacteriophage, right? This is the bacteria that we have, and here you know that it is a bacteriophage. We discuss Hershey and Chase experiment for it, right? So what is it? It is the bacteriophage. Bacteriophage is a double-stranded DNA virus. What is bacteriophage? Bacteriophage is a double-stranded DNA virus, right, bache? So now when this double-stranded DNA virus, when it is passing its DNA to this bacteria, and this DNA is also getting integrated here, so obviously this is what? Transduction. So what is transduction? When virus is used for genetic transfer or for DNA transfer, in simple words. Clear? Clear? So these are the means of sexual reproduction in the case of bacteria. So conjugation is there, transformation is there, transduction is there. Okay, transduction is there. Is that clear? Sure, bache? Right? So we are basically passing the DNA with the help of bacteriophage. Actually, this bacteriophage will infect this bacteria. It will incorporate its DNA here. Now this bacteriophage will grow within this bacteria. So when this bacteriophage will start infecting other bacterias, right? When it will start infecting other bacterias, so obviously in this way there will be the genetic transfer, right? So when viruses are used for the genetic transfer, for the DNA transfer, this is transduction. So bache, in detail in class 12 chapter molecular basis of inheritance, we discuss these experiments in detail. Okay, we discuss these experiments in detail. So this is about the sexual reproduction in the case of bacteria. Is that clear? This is about the sexual reproduction in case of bacteria. And yes, bache, how can I forget? So I hope nitrifying iron, hydrogen, these examples are clear to you. So sulfur bacteria is a chemosynthetic bacteria. Green sulfur bacteria is a photoautotrophic bacteria. Green non-sulfur bacteria is what? Green non-sulfur bacteria is what? It is basically the chemoautotrophic bacteria. So green sulfur, it is photoautotrophic, right? So it will use the pigments. It will use the pigment. I told you, no, some pigments are there in bacteria also. And moreover, guys, can you quote the example of parasitic bacteria, symbiotic and saprophytic? Saprophytic, which will feed on dead and decaying organic matter. Symbiotic, which live in mutualism. Both are getting benefited. Parasitic, you know that. Some bacteria which are present in our system, endoparasites, right? But symbiotic mein the best example is of rhizobium. Do you remember rhizobium? In the root nodules of leguminous plants. In the root nodules of leguminous plants. Right? We have this now. In the root nodules of leguminous plants. Clear, bache? Clear, bache? Saprophytic mein we have many examples. We have many examples. Parasitic mein we have many examples. Pathogenic, non-pathogenic. Too many examples are there. Okay, so what is your homework? This is what you need to mention in the comment section of this particular lecture, right? So homework is, you will tell me about the useful, you will tell me the name of useful bacteria in the comment section. At least 3 examples, minimum 3 examples bache, and harmful bacteria, right? Even mention the disease there. 3 examples each. Okay, 3 examples each. Is that clear? Right, 3 examples each. That is your homework. Okay, so this is about the Monera. I already told you about the shapes. This is your second homework, right? This is your second homework. This is what you need to tell me. Okay. So second homework is, you have to quote the examples here. The examples of spherical bacteria, cylindrical bacteria, spirillum or vibrio. Again, this is your homework. Fine. The next part is, okay, different type of bacteria. So firstly, we will start with the archaebacteria. Right, bache? We are going to start with the archaebacteria. So I have already explained you this, isn't it? I have already explained you this, isn't it? So when we talk about the archaebacteria, it is the ancient bacteria. It is the primitive bacteria basically. It can survive in harsh conditions, right? It can survive in harsh conditions, means in extreme conditions. In extreme conditions, like let's say the temperature is very high, the pH is very low, the pH is acidic. Let's say the salt concentration is very high. Okay, so archaebacteria is the primitive bacteria which can survive in harsh conditions, or you can simply say in the extreme conditions where we consider the life is not possible, right? So wherever we consider the life is not possible, you will find the bacteria there, the archaebacteria, right? The archaebacteria. So we have 3 categories: halophiles, which can survive in the extreme saline conditions where salt condition is too high, right? Then it is thermoacidophiles. Thermal means temperature, where temperature is high. You can say that near about 80 degrees Celsius or above that. Acidophile, where even pH is very low. Let's say pH is 2, like hot springs. Methanogens in marshy areas which produce the methane. So these are 3 categories, right? These are 3 categories. Are you getting my point? Archaebacteria. Are you getting my point, bache? Archaebacteria, ancient bacteria. It is, right, it can survive in the extreme conditions. So I have given you the reason here, right? So you know that their cell wall lacks peptidoglycans, right? Their cell wall, it lacks peptidoglycan, and you can say that muramic acid. Right? The lipid cell membrane is monolayer. Cell membrane is monolayer of lipids, right? And here you will be having the branched lipids. Are you getting it? Yes, bache. So because of that, there will be less fluidity. So it's a chance to survive in the extreme conditions, isn't it? Okay. And in the archaebacteria, ribosomal proteins. Usually ribosomal proteins are basic, but here they are acidic in nature, right? They are acidic in nature. Clear, bache? So there are differences in the structure which help these archaebacteria to survive in the extreme conditions. Fine, bache? To survive in the extreme conditions. So there are 3 examples that you need to remember: halophiles, methanogens, thermoacidophiles, right? Halophiles, methanogens, thermoacidophiles. Clear? Thermoacidophiles. So I hope the meaning of these terms is clear to you. So methanogens, that will produce methane. right? Methane. Methane is also the part of the biogas, na? Gobar gas. Methane. Methanogens. So these methanogens are even present in the stomach of ruminants. You know, na, ruminants like cow and buffalo, right? Like cow and buffalo. So their stomach is also having the ruminants. They are ruminants basically, because stomach is in, different parts are there in stomach. Rumen is also there. The pouch like stomach is there. So here we will have the methanogens which produce the methane gas. So the dung of these ruminants, right? The dung of these cow and buffalo, it is used for the biogas production. Okay, because that dung is the source of methanogens. So these methanogens, they are what? They are anaerobic. Right, they don't need oxygen for their survival. Methanogens. Is that clear, bache? We will even revise it from NCERT. Tell me, is that clear? Is that clear? So basically, what will they do? They are going to form the methane. Okay, they are going to form the methane. Is that clear? Yes or no? Quick. So they are what? They are anaerobic. Right. So they are obligate anaerobic, strictly anaerobic. They cannot survive in the oxygen, in the conditions where oxygen is present. Fine? Sure, bache? Sure? Okay, so next is halophile. So I told you already the meaning of halophile. So when it is a halophile, they can survive in extreme saline condition. Right, they can survive in extreme saline conditions. So halophiles, we have the example of halobacterium. Right, we have the example of halobacterium. So these bacteria can even form a kind of layer around them. When they are living in extreme saline conditions, they form a kind of layer around them which can trap the solar energy. They can use that solar energy for the ATP formation. Okay, they can use that solar energy for the ATP formation. But in the case of methanogens, they are chemoautotrophs. They are not going to use the sun energy at all. They are chemoautotrophs. Clear, bache? And when it comes to the thermoacidophiles, I told you already, where temperature is high, where pH is low, like you can say that pH is near about 2. So in such high temperature, low pH conditions, some bacteria they can survive. So we have the example of your thermoacidophiles, right? So thermoproteus, that type of examples are there. Right, that type of examples are there. So this is about the archaebacteria. Clear, bache? This is about the archaebacteria. Is that clear? And okay, so after it, mycoplasma. We are talking about the different types of bacteria now. So next is mycobacterium. Okay, or you can say that mycoplasma. So bache, what do you know about this mycoplasma? What do you know about this mycoplasma? Do you know anything about the mycoplasma? Ma'am, meaning of obligate. Obligate means strictly, like obsessed to something. That organism that need that particular thing only. Like you are saying that no, for biology we need Ambika ma'am only. Right? We cannot study from anyone else. Right? So strictly you want me only. You are obsessed for me. Obligate will be the word that we will use there. So mycoplasma, it is the only bacteria which is without cell wall. Right? It is the only bacteria which is without cell wall. Right, bache? Right, bache? And it is a facultative anaerobe. Means it can even survive. Although it lives in oxygen, but it can even survive without oxygen. Are you getting my point? It can even survive without oxygen. Okay, so mycoplasma, the only bacteria which is without cell wall, it is a facultative anaerobe. Now, it is without cell wall, so obviously it is not having a proper shape. Cell wall is going to provide it a shape, na? So it is not having a, right, it is not having a proper shape, isn't it? It is not having a proper shape. So when bache we see the structures where the proper shape is not there, like let's say this is a bacteria which can change its shape. So for such organism we use the word pleomorphic, right? We use the word pleomorphic. pleomorphic, right? Pleomorphic means having different shapes. What is the meaning of pleomorphic? Pleomorphic means having different shapes. What is the meaning of pleomorphic? Pleomorphic means having different shapes. It is having what? It is having the different shapes. Clear bachche? Clear bachche? So it is pleomorphic. It is pleomorphic. Right? Right? So this is pleomorphism. So because mycoplasma is not having a fixed shape, it can have different shapes. So it is also known as jokers of microbiological park. Right, bache? We even call it as joker of microbiological park. What is it? These are the jokers of microbiological park. Okay, so only bacteria without a cell shape, facultative anaerobe, it shows the pleomorphism, and if you remember, it is the smallest living organism, right? It is the smallest living organism, that is 0.3 micrometer. And among that, it is the PPLO, pleuropneumonia-like organism, which is 0.1 micrometer, right? Which is 0.1 micrometer. Is that clear, bache? Which is 0.1 micrometer. So basically, we are talking about what? We are talking about the mycoplasma. Clear, bache? Clear, bache? Yes? Okay. Done. So because it is not having the cell wall, so those antibiotics that destroy the cell wall, they cannot affect it, isn't it? They cannot affect it. So it is protected from antibiotics like your penicillin, like your lysozyme. So it is protected from antibiotics which destroys cell wall, right? Which destroys cell wall. Okay, mycoplasma. And this mycoplasma is non-motile. It is not movable. It doesn't have flagella. Clear, bache? Clear, bache? So it is protected from antibiotics which destroys cell wall, but you know, tetracycline that affects metabolism can destroy it. I'll explain it, just write down the point as of now. See, bache, it's very simple. Now we have a bacteria which is not having the cell wall. So obviously any antibiotic which kills that bacteria by, you know, hydrolyzing the cell wall, by not letting the cell wall to grow, is not functional on the mycoplasma. Like if, let's say, if I have any disease, right, which is from mycoplasma, and I am taking the antibiotics which acts on the cell wall, right, which functions by destroying the cell wall. So obviously it will not be functional over mycoplasma because it is not having the cell wall, right? So penicillin acts on cell wall, so it cannot affect, it cannot do anything to the mycoplasma. But we have tetracycline, we have another antibiotics also. Some antibiotics, they break down the cell wall. Some antibiotics, they stop the protein synthesis. Some antibiotics, they don't let the ribosome to bind to the mRNA, right? So there are different ways by which an antibiotic stops or kills the bacteria. Okay, by which the antibiotic stops and kills the bacteria. So here the tetracycline, because it is going to stop the metabolism, so it can destroy this mycoplasma, but your penicillin cannot. Okay, bache, but your penicillin cannot. Is that clear? Is that clear? Yes or no? Yes or no? Okay, so mycoplasma, it's a very simple bacteria, only bacteria without cell wall. So its structure is very easy to understand, but its behavior is very complex. Okay, its structure is very easy to understand, but its behavior is very complex. Fine, its behavior is very complex. And as I said, they are the jokers of microbiological park, but along with them, we also call them as jokers of plant kingdom. Right? Mycoplasma is also known as jokers of plant kingdom. They are also known as what? Jokers of plant kingdom. Now you must be thinking, ma'am, why jokers of plant kingdom? Because they are, initially they were placed in the plant kingdom. Okay, initially they were placed in the plant kingdom, right? You got it, bache? Is that clear? Sure? Okay. So we are done with the archaebacteria, we are done with the mycoplasma, right? So now we have to discuss the photosynthetic bacteria, that is your cyanobacteria or the actinomycetes, okay, or the actinomycetes, okay. Fine, fine, fine. Done. So let's talk about the cyanobacteria. Also known as photosynthetic bacteria, also known as blue-green algae. Blue-green algae. Bache, can you quote any example of a photosynthetic bacteria? Can you quote any example of a photosynthetic bacteria that is your blue-green algae? Yes, blue-green algae. Tell me. And bache, this mycoplasma, the cyanobacteria, these actinomycetes, they all are the example of eubacteria. Like in the bacteria, we discuss it in two categories, na, archae and eu. So archaebacteria, I told you, methanogens, thermoacidophiles and your halophiles. And if it is eubacteria, cyanobacteria, your mycoplasma, your actinomycetes, they will come there. So don't get confused here ever, right? Don't get confused here ever. So now we are discussing what? We are going to talk about the cyanobacteria, right? They are going to talk about the cyanobacteria. So mostly they are gram negative, right? Mostly they are gram negative, right? And even they are oxygenic. They are what? They are oxygenic, means they will, during photosynthesis, it produces oxygen. It releases oxygen, okay? Clear? It releases oxygen. Cyanobacteria. What is it? It's a cyanobacteria. So the example that we read in the NCERT, the best example is nostoc, right? Remember that filamentous structure? In the case of nostoc, we used to see some filamentous structure there, right? And that filamentous structure is covered with the mucilaginous sheath. Remember, that filamentous structure is covered with the mucilaginous sheath. And here in this structure, we will even discuss some different cells also, right? We even talk about some different cells also, isn't it? Isn't it? That is what we discuss, yes or no? Chemosynthetic bacteria, blue-green algae, bache, we are discussing, right? So now, is that clear? Up to this part all clear? Up to this part all clear? Yes? Okay. So they are also come under the Myxophyceae. We even put it in Myxophyceae, these photosynthetic bacteria. Now, bache, when you look at its structure, right, just look at its structure very very carefully. No doubt it is unicellular, but sometimes some cells together, like here in the case of nostoc, you will see many different types of cell. Together they are forming some filamentous structure. Together they are forming some filamentous structure. So they are unicellular, they can be filamentous, and they can be colonial, right? They can be colonial. Are you getting my point? Like here in the case of nostoc, all the cells are not same here. In the case of nostoc, you will also see some different type of cells, and we used to call it as heterocyst. So here in the case of photosynthetic bacteria, this is the heterocyst that is very important to understand, to know, okay? Okay, so this is what we need to understand, this is what we need to note down. So cyanobacteria, the structure can be unicellular, filamentous, colonial, many single cell, they live together, right? They live together, they form filamentous structure, right? They form filamentous structure, right bache? Right bache? So structure is just like a typical bacteria. Their structure is just like a typical bacteria, right? Right, it is just like a typical bacteria, like there is no nuclear membrane, right? Like this it will be. But here, what is special about these? What is special about these cyanobacteria, photosynthetic bacteria, blue-green algae? Bache, they are self-dependent. Right? This bacteria is self-dependent. It can fix, here we will see the example, which can fix nitrogen, atmospheric nitrogen, and can perform photosynthesis. Right? And can perform photosynthesis. Are you getting it? It can fix nitrogen and it can perform photosynthesis. Okay. Okay, so they are self-dependent, and you will see all the cyanobacteria, they all are non-motile. You will not see any motile structure here. They all are what? They all are non-motile. They all are what? They all are non-motile. And in their case, you will see some, sac-like structures. I was explaining you now. I was explaining you, bache, when I was explaining you the structure of bacteria, I used the word chromatophore. I use the word chromatophore there. Chromatophores there. Some pigment containing sac-like structures. So that pigments will help in the photosynthesis. Okay, that pigments will help in the photosynthesis. And moreover, bache, in the photosynthetic bacteria, you will even see some example, like they are having some thylakoid-like sac-like structure. Do you know the word thylakoid? Thylakoid, in the case of chloroplast, we discuss it now. So some thylakoid-like structures, some thylakoid-like lamellae are also present in the case of photosynthetic bacteria, right? Some thylakoid-like lamellae are also present in the, right, photosynthetic bacteria. This is what you need to remember. Okay, bache? Okay, done? Done. Now we have to talk about this nostoc. But in the case of nostoc, all the cells, they are not same, right? All the cells, they are not same. Like we will be having some different cells also. It is not mandatory that if there is any filamentous algae or filamentous blue-green algae, so they will have all the cells, same type of cells. No. Like in the case of nostoc, you will see the heterocyst. So bache, heterocyst is very important to understand here. Now, when we take the example of nostoc, it can fix atmospheric nitrogen. It can even fix atmospheric nitrogen, right? And it is photosynthetic as well. So these heterocysts, they are linked to the nitrogen fixation. These are the cells which will help in the nitrogen fixation. Now, what is the point here? Actually, here in this filamentous algae, what is the story, bache? Rest all cells, like here, you are having cells. They are photosynthetic. They can perform the photosynthesis. They are aerobic. They all are photosynthetic. They can perform the photosynthesis. They can release the oxygen. But when you talk about the heterocyst, I just told you it is linked to the nitrogen fixation. But this heterocyst is anaerobic. This is something very important. Here in this heterocyst, oxygen is not present here. Right? Oxygen is not present here. It is anaerobic. Why is it so? Because in the heterocyst, photosystem 2 is absent. Right? Photosystem 2 is absent. This is a very important point. Those who are in class 11th, might be they don't understand it. For understanding this photosystem 1 and 2, you need to read the chapter photosynthesis. But I am giving you the overview here. Photosystem 2 is nothing. It is just a protein complex. It is present, right? It is present in the chloroplast. It is present in that thylakoid membranes. Right, it is present in that thylakoid membranes. This photosystem 2 is related to the it is related to photosynthesis, of course, and it is responsible for the oxygen release. Like it is having some oxygen evolving complex. It helps in that oxygen release. Now, we are talking about the heterocyst. So, heterocyst is responsible for the fixation of nitrogen. This is again very important. This is again something important. So, note down. So, it is anaerobic. Anaerobic means in heterocyst, it is without oxygen. Oxygen is not there. Why oxygen is not there? Because photosystem 2 is absent. If there will be photosystem here in heterocyst, oxygen will be released. Right, but we don't want oxygen in heterocyst. We don't want oxygen in heterocyst. What is the reason for it? Heterocyst contains an enzyme nitrogenase. Heterocyst, it contains an enzyme that is nitrogenase. Nitrogenase is nitrogen fixing enzyme. It is used for nitrogen fixation, right? Nitrogenase, it is used for N2 fixation. We need it for nitrogen fixation, and this nitrogenase, it works in an aerobic condition. Now see, in the nature everything is so beautifully adapted, isn't it? So when you talk about the heterocyst, it contains nitrogenase. It is anaerobic. Why? Because nitrogenase cannot work in the presence of oxygen. If there will be oxygen, nitrogenase will not be able to fix the nitrogen. Nitrogenase will not be able to fix the nitrogen, so it works in anaerobic conditions. So the PS2 is absent, and it will help in what? It will help in the fixation of nitrogen. Is that clear, bachcho? Is that clear? So these are large cells, heterocyst. Okay. Okay. Done? Clear, bachche? Clear, bachche? Is that clear? So nostoc, anabina, right? So heterocyst. So this is the nostoc. It is also covered with the mucilaginous sheath. Always remember this. You know, mucilaginous sheath which can hold the water, right? So heterocyst is basically for the nitrogen fixation. So can I say that this cell is not non-photosynthetic here? It is non-photosynthetic here. So heterocyst is non-photosynthetic. Rest all cells are photosynthetic, right? Rest all cells are photosynthetic. So we have examples of, you know, many photosynthetic bacteria. So nostoc, your anabina, right? They all are having the heterocyst. And it is not mandatory that all the cyanobacteria will have heterocyst. Like there are some homocystous blue-green algae also, like your oscillataria, right? It doesn't have the heterocyst. So it doesn't have the heterocyst means it will play no role in the nitrogen fixation. Clear? It is not going to play any role in the nitrogen fixation. It will just fix the, it will just do the photosynthesis, okay? It will just do the photosynthesis, okay? Done, bachche? Fine. So this is about the blue-green algae, photosynthetic bacteria. So heterocyst is non-photosynthetic, nitrogen fixing. So these points are very, very, very important. Clear, bachche? Clear, bachche? So also remember, in blue-green algae, right, cell membrane form lamellosomes. Sometimes, you know, you find this word in the questions, lamellosomes, right? So in blue-green algae, cell membrane form lamellosomes. They are nothing. These are the structures just like mesosomes, right? These are the structures just like mesosomes. That's all. But I will share the PDF with you all. But yes, it's a request. Please note down all the important points. Okay, please note down all the important points. But here all the cells are vegetative. It's just that few are photosynthetic, few are non-photosynthetic. That's all. That's all. Okay. Done. So let's revise it from NCERT, but before that, see these tables. So here I have added, like, some are photolithotrophic and autotrophic bacteria na. So energy source is light, hydrogen, electron sources, you know, inorganic H, fine. And here photo-organotrophic, light, organic, electron donor is there. So bache, you can use these notes after the class, right? Right, you can use these things and you can revise the chapter. And now we will revise the kingdom protista from the kingdom monera from your NCERT. Just a minute. Where is your NCERT? Okay, I don't have your NCERT here. I do have NCERT, just a minute bache. Okay, so see, this is the kingdom monera. Okay, so kingdom monera, bacteria are the sole member. So they are most abundant, they are present everywhere. So you know na, some bacteria are useful as well. Some bacteria are useful as well. Very useful they are. I'll give you one example. Although there are so many examples. So useful bacteria may the best example is of lactobacillus, which helps in the curd formation. Right, lactobacillus, which helps in the curd formation. We have Acetobacter aceti, which will help in the formation of acetic acid. Remember, microbes in human welfare. Propionibacterium shermani, which will help in, yes, it will help in cheese formation. So bacteria, they are present everywhere, right bache, they can survive in extreme habitats also, right, even in the hot springs, in the deserts, in the snows, in the deep oceans, right. So bacteria are grouped under 4 categories. I told you about the shape, the coccus, bacillus, the comma shaped, and the spirillum, right, and the spirillum. And then bache, its structure is very simple, but they are complex in behavior. Okay, so done. So archaebacteria, they are special, they live in some of the most harsh habitats, right bache, like extreme salty areas, halophiles. I gave you the example of halobacterium. In hot springs, thermoacidophile, thermoproteus is the example. Marshy areas, methanogens, methanobacterium is the example, right bache. So they differ from others in having the different cell wall structure. I told you what is different in the cell wall of the archaebacteria, right bache. So they can survive in extreme conditions. Methanogens are present in the ruminant animals, in the ruminants of the cow and buffalo. I told you about it. And then we have the eubacteria. So bache, eubacteria will include all the examples like your blue-green algae, actinomycetes, mycoplasma. They all come under the eubacteria. Okay, they all come under the eubacteria. Clear bache? Done? So see, photosynthetic bacteria, photosynthetic autotrophs. So bache, even in photosynthetic autotrophs, there will be the chlorophyll A, just like the higher green plants. Okay, just like the higher green plants, right, just like the higher green plants, they will also have the chlorophyll A. Clear? So cyanobacteria, they are unicellular, colonial and filamentous. They can live in fresh water, marine or terrestrial, right. So this is not, see, we are using the term algae, but it is not the part of kingdom Plantae. These are blue-green algae, photosynthetic bacteria, cyanobacteria. So don't get confused here bache, right, don't get confused here. So colonies are generally surrounded by gelatinous sheath or mucilaginous sheath, right. They can even form blooms in polluted water bodies. Okay, and some of them can fix the nitrogen. So nitrogen will only be fixed by those blue-green algae which are having the nostoc, remember, which are having the heterocyst, remember it. So if any blue-green algae is having the heterocyst, means it can fix the nitrogen, right. So if it doesn't contain heterocyst, it cannot fix the nitrogen. Like your oscillataria, that is just a photosynthetic, bacteria that cannot fix the nitrogen. Okay, that cannot fix the nitrogen. Clear bache? So chemosynthetic autotrophs bacteria, they can oxidize various inorganic substances like nitrates, nitrites, ammonia. So with the help of that table, you guys can revise it. So right, so they will use energy for the ATP production, they will help in the recycling nutrients bache, right. So heterotrophic bacteria are the most abundant. We have the decomposers bache, right, and they are having different roles also. So rhizobium bacteria, the best example of symbiosis. It lives in the root nodules of leguminous plants and it will help in the nitrogen fixation. Clear bache? So this is your homework. Like see here, the disease causing bacteria they are given, a cholera, typhoid, tetanus, citrus canker. That's your homework, right? Definitely question will come from this part, but that's your homework. You have to mention it in the comment section. You have to mention it in your notes. Like cholera is caused by Vibrio cholera. Typhoid is caused by Salmonella typhi. Tetanus is caused by Clostridium tetani, right? Your citrus canker is caused by, I forgot. Citrus canker is caused by, yes. It is also a bacterial disease, that I'm sure, but I don't remember the name right now. So this is what you need to write down, right? This is what you need to include in your notes. This is important. Okay, it is important. This chapter is literally lengthy, bache. Right? No doubt you have to follow the NCERT for this particular chapter, but at least you should know about the things in detail. Okay, then the revision from NCERT will be easy for you. Fine? It will be easy for you. So this is what you need to do. The citrus canker is caused by, this is what you have to tell me in the next class. Fine? Fine. And it's a, let me tell you, it's a plant disease. It's a plant disease. Right? So bacteria, you know that it can reproduce mainly by fission, but in unfavorable condition, it can go for the spore formation. So spore formation means sporulation. So endospore, internal spore will form. Clear bache? Clear bache? And here they are talking about the sexual reproduction na, by adopting a primitive type of DNA transfer. So here they are talking about the conjugation, transformation and transduction. Right? So we have discussed everything written in your NCERT. Clear? Clear, clear, clear? Yes. So mycoplasma, done. So here you can see the nostoc, the heterocyst is present here. So here the nitrogen fixation will be done, and here also bache you can see the anabina. This is what I have added. So see here, these are the heterocysts. These are the here in this anabina also it is also filamentous. Some cells are having large size, right? Some cells are having large size. So that is basically the heterocyst. Clear bache? That is basically the heterocyst. So this is all about the kingdom Monera. Here you guys can also see this bacterium dividing. So first step will be your DNA duplication, then your mesosome will grow, right? Mesosome will grow, cell membrane formation will be there, your mesosome will divide and then cell plate will form. Right? So it is going to be very simple. You will not discuss the prophase, metaphase, anaphase and telophase here. Clear bache? So these type of things are not there. So now we will keep one more part for it, right? In second part we will finish protista, fungi, viruses and all. See bache, citrus canker, it is citrus canker. Yes, you are right. Bacterium Xanthomonas. It is. Grace, very good. The name of the bacterium is Xanthomonas. So bache, in the next class we will revise the protista, right? I will keep part 2 for it. So we will do the plant kingdom later. So we will finish the what are we going to finish? We are going to finish the protista and the fungi and the viruses part there. Okay? Okay? Because see, we don't want to rush. I can finish it in 4 hours, in 5 hours, in 6 hours also, but we don't want to rush. Fine, we don't want to rush. So that is why I will take it in 2 parts, right? But not more than 2 parts. So next class also will be for 3 to 4 hours. But yes, we will finish the protista. Very simple, very easy it is. And fungi and then the viruses and all. Okay, so do revise it, and tomorrow at 9 PM I will take a practice session so questions will be there from the chapter Living Word and from the biological classification till Kingdom Monera. Okay? So if you have any other doubt, just let me know. Right? If you have any other doubt, just let me know, and I will share the PDF in the Telegram group, but I will share it after test. Okay? I will share it after test. Not today, after test. Thank you, Soumyadeep. Okay, and guys, I want to see the comments in the comment section, right? You don't write anything. Please, whatever questions I'm asking now, whatever is your homework, please mention all that answers there in the comment section. Okay, so see you tomorrow. So tomorrow at 9 PM there will be the problem discussions. There will be the problem practice session. Okay. So bye-bye, Tara. Thank you so much. Good night. Do revise it from NCERT, and please start making the short notes. It will help you out. And in NEET Conquer batch, this is how we are going to teach. Now you guys can see, and the same way we are teaching you in the NEET Energized batch or in the NEET Avengers batch. The only difference is there we are taking a lot of tests, very frequently the doubt solving is there, or you can say that quick revision sessions are there. Okay, so if you are serious for your NEET examination, start preparing properly. If you can join any batch, please be the part of a batch. Okay, so in a disciplined way, you will be able to cover your syllabus. So thank you so much.